speaker, Dr. P.V. Baraprasad. So to introduce him, let me share one slide. So hope the slide is visible. So Dr. P.V. Baraprasad, he is presently Professor of Crop Ecophysiology and Director of Center for Sorghum Improvement in the Department of Agronomy at Kansas State University. Regarding sir's education, he has completed his graduation from Angrao University in Hyderabad. Regarding sir's research area, so it is understanding responses of food grain crops to changing environments, that is temperature, water, and climate change factors, and developing crop management strategy and efficient use of inputs. Sir has also quantified impacts of abiotic stresses on several grain crops. Then he has improved the understanding of mechanisms associated with several abiotic stress tolerance. Sir has a very good credit for identifying physiological tools to determine drought and high temperature tolerance, especially in wheat crops. Then he has screened germplasm collection of several crops like wheat, sorghum, then several legumes, coffee, etc. for tolerance to drought and high temperature stress. Sir has developed extended sustainable crop production practices for improving resource use efficiency and productivity of grain crops in several crops. So he has a role in these crops across the country, across the globally. He has been involved as an advisor for several projects and all. Regarding fellowship, he is the fellow of American Association for the Advancement of Sciences, then Crop Science Society of America, American Society of Agronomy. Sir has been on editorial board of several journals. I'm not speaking into the details. Then regarding scholarship and awards, there are more than 25 awards to sir. I will name few of them. So he has received Irvin E. Youngberg Award, then Martin and Ruth Massengal Lectureship, then Outstanding Scientist Award, Distinguished Faculty Award, then IMFR Excellence Award, then Graduate Teaching Award from Kansas State University, then International Educator Award. He is very much associated in the guidance and planning of the international education for several countries' higher education. Then Outstanding Agricultural Scientist, then Early Career Award, and Young Scientist Award. As far as teaching is concerned, so as a major professor, sir has guided 23 graduate students, including 9 masters and 14 PhD students. He has trained more than 40 scholars from 15 countries across the world who are holding prominent positions in different countries. So I have been also mentored by sir at Kansas State University during 2013-14. So thank you, sir, for giving the opportunity to me and imparting knowledge on abiotic stress screening and all. Then regarding project funding, so sir has good success rate. Sir has received more than 145 project funded by him. So there was good success rate. More than 90% projects were funded to sir. Then regarding publication, more than 149 articles of various national and international journals of high repute has been published by sir. Then which included then he has also published more than 230 abstracts in different conferences and symposia, more than 70 strategic reports and brief reports. Then he has made more than 100 presentation in different forum at conference as well as different dialogues symposium. And sir has been invited for talk for more than 70 lectures by different countries at a different level. So I'm not going into the much detail. Sir has been involved in several things across the globe, teaching, guidance, then collaborative projects, etc. So thank you very much, sir, for accepting our invitation to talk to our training participants about the abiotic stress, their impact and challenge. So with this, I would like to invite Dr. P.V. Baraprasad for presenting his talk. I'm stopping my slides. Thank you very much, sir.
Are you able to see my slides?
Yes, sir. Yes, sir. Uh, Sir, please do in presentation mode, sir. Okay.
Yeah. Okay. Do you see it in presentation mode now or no?
Yes, sir. Yes, sir. Not now, sir. Just.
Second, yeah.
No,
It's in
sir.
the
It is.
presentation mode now?
No, sir. It is not in presentation mode.
Can you see it now?
Still not, sir. It is still in.
Still not in presentation mode?
Not, sir. Maybe, sir, resharing.
Yeah, let me do it now. Still not in presentation mode?
No.
Okay, let me stop and do it again.
Yes, sir. Yeah, stop and resharing, sir.
Still not? Still not in this, still not in the presentation mode?
No, sir. It is in like that slide view. With all sides are being shown. Click on,
Okay,
sir.
I have three screens, so hopefully.
Um,
Sorry about that. I'm swapping it, it's still not showing.
is a slide sorter view.
Yeah, it's full, it shows on my full screen, but
Okay.
I'm not sure what's happening. All right, can I change the computer 1 second?
Okay,
Should
sir.
be.
Okay. No issue,
It
sir.
should be. I'm surprised it's not showing on that. So
Okay, sir. Okay, sir. Yes, sir. No, it's
Yeah,
fine, sir.
it's okay, sir.
Yes, sir. Fine, sir.
Okay, can you see full screen now?
Yeah. Yes, sir. Yes, sir.
Yeah, I'm so sorry. It never happened. It's a new computer, but I thought some reason there's four screens and I was not able to manage it. So I
Yes, sir.
apologize
No
for
issue,
that.
sir. It happens, sir, sometimes.
All right, good. So
Mhm.
I'm glad that you're able to see the full screen now.
Yes.
Thank you very much, Jyoti, for giving me this opportunity and for your kind introduction. So let me get my timer started. So in the next an hour or so, I'll be talking about this topic today, abiotic stress and crop yield. And first, we'll look into the impacts, evaluation, management, and opportunities. And I'll be watching the screen. So if anybody has any questions, any point of time, please don't hesitate to ask right away. So this will be the outline of my presentation. First, I'll talk about climate change, what has happened in the past, and then what is the current situation and how the future scenarios will look like. And then we'll quickly go through the overview of drought and high temperature stress, what happens if these two occur at different stages of crop growth and development. And then I'll talk about the impacts of short duration of temperature stress and drought stress. And then quickly about the longer duration of heat stress. And then what are some of the thresholds for major grain crops. And then we'll look into how do we evaluate larger germplasm collections, important traits and screening methods, and then both physiological and high throughput methodologies. And then what options do we have in terms of managing the stress? We will look into quickly both agronomic management and also some of the breeding techniques. And then I'll quickly go through the what type of plant genetic resources we have, and is there any genetic variability existing for both drought and heat stress? And I'll give you specific examples for 4 different crops: rice, wheat, sorghum, and soybean. And then we'll conclude and we'll have some time for questions. So I'll finish it up in the next hour or so, but I know we started 10 minutes late, so I'll try to do it quickly. So first, let's look into what's climate change and what has happened in the past, and what is the current situation, and what is the future scenarios. If you look at the greenhouse gas emissions, they have significantly increased from the beginning of last century. But I'm showing you here the data for carbon dioxide from 1960s onwards up until at this point of time. In these 4 visuals, you have the carbon dioxide emissions, and then you have the methane emissions, and you have the nitrous oxide emissions, and you have sulfur hexafluoride emissions. So most of these greenhouse gases have significantly increased over a period of time. And at the present time, the carbon dioxide concentrations last year in December were about 417 ppm. That has been the highest so far, and it is going on at this stage. We will be following the recommendations or following the projections of the IPCC. And what it has done over the last several decades is you have 2 visuals here. The left one is showing you the average temperature increase change in the last 50 years compared to what the baseline from 1951 to 1978 was. As you can see, most of the regions in the world has significantly increased the temperature, anywhere between 1 to 2 degrees Celsius. And on your right-hand side is a visual of the global average temperatures from 1850s to all the way to 2020. And this is a global temperature anomaly, or the difference in degrees Celsius. And if you look at what was in 2021 relative to the last century, the temperature has risen by about 1.2 degrees Celsius. So these are significant increases. And most of you are aware of what happened in the Indian subcontinent last year. Here I'm showing you the daily maximum temperature in the summer from March to April 2022. That was probably one of the hottest year ever recorded for Indo-Gangetic Plains. Most of the country was extremely hot, and you can see that during that time the average temperatures, daily maximum temperature, what it was. If you look at in India in April, in Uttar Pradesh, it reached almost 47.2 degrees Celsius. It was a record. And there are also several other places in Gurgaon, Chandigarh, and Dharmshala where the temperatures were extremely high. And similar was the situation also in Pakistan and also rest of the India, so to speak. So on your right hand side is the anomaly of 2022 compared to that of what the average temperature was from 1979 to 2022. So it was almost in some places, majority of the areas, it was anywhere from 2 degrees Celsius to 5 degrees Celsius warmer. And this has significantly impacted the wheat yields. And when it comes to precipitation, which is a combination of the rainfall and snowfall, same time, March and April, the Indo-Gangetic region was extremely dry. And this visual shows you the deviation from the long-term average of precipitation from 1981 to 2020. And as you can see, in some places, the deviation was almost 20% to 100% less precipitation. So both of these temperature stress and extreme precipitation or the rainfall stress has significantly impacted. How does the future look like? These are the different models projection of air temperatures and top two visuals depending upon what type of scenarios you have high climate sensitivity versus low climate sensitivity. But either way, any of these models, if you look at it, they project the temperature would, if you go business as usual, then the temperature will increase anywhere from 2 to 5 degrees Celsius. Whereas if you look at the projections of the precipitation, it is highly variable. In some parts of the world, especially in southern part of Africa and South America and several other places, you might see a decrease in the precipitation. But overall, they project that the rainfall will increase. But that rainfall will fall in short period of time, or the intensity of the rainfall will be high. The amount of rainfall which you receive in a year might be received in a very low number of rain events. And also combining with the high temperatures, then even though you might have higher rainfall, but you'll still have, because of the higher evapotranspiration demand, the soil moisture available to the crop will still be less. So in future scenarios, there will be two projected that not only the increase in the average temperatures, but also you are more likely to see the occurrence of extreme events, meaning that extremely hot or hot weather, especially when it comes to temperature. And we all know that if these extreme events, even though if they are of a short duration, anywhere from 1 to 2 degrees, 2 days, it might still be able to negatively impact the productivity of the crops, especially if these events happen during the sensitive stages of crop growth and development. So that's the scenario of the climate change, or particularly in terms of temperature and precipitation, what has happened in the past, what is happening now, and how it looks like in the future. Now we'll look into the impacts of high temperature stress and drought stress, and what are the major processes in terms of yield process and the components of yield and how they impact it. So first, I'll go through in a generic view, and then we'll dig a little bit deeper. But to begin with, I just wanted to make sure that all of you are aware of crop growth and development. What is the difference between the crop growth and what is development? Growth is the increase in the size, volume, or height of the plant, whereas development is referred to as change in the growth phases, like vegetative stage to reproductive stage to physiological maturity. And within those phases, there is multiple stages. In this visual, I am showing you the developmental stages of sorghum. Those range from 0 to 9, and then the growth stages from emergence to 3 leaves to 5 leaves, the growing point differentiation, and the flag leaf, boot leaf, half boom, soft dough, hard dough, and physiological maturity. And these are the different developmental stages. Not all of the stages are equally sensitive to temperature or drought stress. Some of them are relatively more sensitive than others. And here is a crop growth and developmental stages of soybean. In this case, we call emergence the first trifoliate, second trifoliate, and we go through all the vegetative phases. And then R1 is the beginning of flower, full bloom, beginning of pod, full pod, beginning of seed, full seed, beginning maturity, and physiological maturity. So that's a legume. So I just wanted to give you a feel for both cereals and legume. And again, as I said, not all stages of crop are equally sensitive. In general, for most of the grain crops, whether it is cereals or legume, reproductive stages of crop growth and development are much more sensitive, particularly flowering and few days before flowering and anthesis, and also at after the seed set during the grain filling duration. I'll come back and talk a little bit deeper about those things. So first, I'll give you an overview now. And here I'm showing you a cereal of sorghum. So if you have, whether it is temperature stress or drought stress, if you have the stress after emergence or sowing to the emergence, it'll impact the germination percentage if it is happening before emergence. But after emergence, it impacts the rate of germination. And both of these will significantly influence the plant population. That is one of the major components of the yield. And assume all of you know what are the yield components. So if you multiply your plant population by seed numbers, by individual seed weight, you get the yield. The plant population times seed numbers times weight of the individual grain or seed will give you the yield. So I'm trying to show you the impacts of these stresses and what yield components and how it is impacted. So if you have the stress between sowing and seedling emergence, it impacts these two items and it will impact your plant population. If you have the stress after seedling emergence to vegetatively mature plant, your vegetative development, the vegetative growth, the leaf growth, shoot growth, and the root growth is impacted, which will impact the leaf area, which also the resource capture, especially light interception, and also structure and the biomass is impacted. But if you have the stress after the vegetatively mature plant and before the floral bud initiation, so that's the time when the initial yield potential is being set. So that will set the number of potential reproductive sites. And then if you have if you have the stress between flower bud initiation or flowering and anthesis, it will impact your gamete development, whether it is pollen development or ovule development, especially the viability of the pollen or the ovule is affected. And of course, if there is stress, it also impacts the flower abortion as well. And if you have the stress after flowering and anthesis before the seed set, it will negatively impact the anthodiescence, pollination, pollen germination, pollen tube growth, stigma receptivity, and fertilization is impacted. And that will negatively impact your fruit set or seed set, or fruit, and then eventually it will lead to lower fruit or lower seed numbers. Then if you have the stress after the seed set until the physiological maturity, the things which are happening during those stages are the seed development, the seed filling rate and the seed filling duration, and also seed composition. And generally, as soon as the seed is set, if the stress is happening at the initial stages after the seed set, it leads to embryo abortion and then decreases the seed numbers. But if it happens after the start of the seed filling, then it negatively impacts the seed filling duration generally decreases under high temperature stress or the drought stress, which leads to shorter grain filling duration, leading to smaller seed size, and then eventually that leads to lower seed yields. And so in most of the cases, it also impacts the seed composition, which might negatively impact not only the nutritional composition of the grain, but also the next season viability and the seed quality is also impacted. So that gives you an overview of what stress will do if it happens at different stages of crop growth and development. And then now, if you're looking for the temperature or drought tolerance, what are the things which you might want to look at it if you are screening the germplasm, especially the larger germplasm collections? So if you have the pre-flowering stress, you're looking at those genotypes where the cell growth is not impacted, or the photosynthesis is good, and the biomass is produced well, and there's enough carbohydrate stem reserves, especially carbon and nitrogen. And if you have the stress at the time of flowering, you are looking for plants which have higher gamete viability, greater seed set, greater seed numbers, eventually forming higher sink capacity. And then if you have the stress after flowering, you're looking at traits such as stray green so that it can continue to photosynthesize, and then higher resource mobilization, meaning that moving of carbohydrates and nitrogen from the senesced leaf or stored stem reserves into the sink, and then higher photosynthesis and the seed size is also some of the traits. I'll go little bit deeper into those aspects when I give you specific examples from various crops. So that gives you a generic overview of what happens when the stress occurs at different stages of crop growth and development, and what are some of the items which are looking under those different stages of stress. So now I'll quickly quantify and give you some real experimental data of what even short episodes of high temperature stress, if it was, let's say, 6 hours of high temperature stress, or even one or 2 days or 5 days of stress, would it negatively impact the yield? And then what components of yield it impacts. And before I go there, I wanted to make sure that you all understand the difference between the climate change and climate variability. Those two are different terminologies we often use in the literature. Climate change refers to what is happening in terms of those temperatures or drought stress or temperature extremes or rainfall conditions over a longer period of time, over decades or over centuries. Whereas climate variability refers to changes which are happening within the season or within a year, especially in the crop growing season, if there is occurrence of extreme events, whether it is cold stress or high temperature, whether it is drought or even flooding type of situations. So and in future scenarios, it's very likely that our crops will be exposed to very short episodes of these stresses, where maybe 1 or 2 days or 4 or 5 days, especially extremely cold temperatures or extremely high temperatures, and also high intensity rainfalls. And those will negatively impact if they occur at the sensitive stages of crop growth and development. So the first thing which we want to know is that determine what is the most sensitive stage for these environmental stresses. I'm going to only cover the two stresses, the high temperature or heat stress, and then the drought stress here. So I've given you here 3 different crops, a sorghum on the top left, and then groundnut on the top right, and the bottom 2 visuals are for the rice. And then let me take a little bit of time to explain this visual. So on your x-axis is the timing or the days of at what time relative to anthesis the stress was imposed. 0 means anthesis, negative numbers, 5, 10, 15 is days before anthesis, and the 5, 10, 15 positive numbers are days after anthesis. And on your y-axis is the floret fertility, which is a percentage of control. We have 2 temperatures for sorghum here. Optimal temperature is 30/20, daytime maximum, nighttime minimum, and high temperature or heat stress is 36 daytime maximum and 26 nighttime minimum temperatures. That's a temperature stress. And in this case, the plants were exposed to 5 days of heat stress. So we grew the plant under optimal conditions. And when they reached that particular time, the anthesis 5 days or 10 days, 15 days before anthesis, we exposed those plants to 5 days of heat stress. And then this is the floret fertility of the tagged flowers. So we tagged individual flowers and then followed them whether they were fertile or not. So as you can see, the impact on sorghum, it's a cup-shaped, U-shaped, inverted U-shaped curve. Shows that the maximum reduction in floret fertility was happening when the stress was few days before anthesis, 5 to 10 days before anthesis. That's when you see the maximum reduction in the floret fertility for 5 days of stress. And then after the seed set, which happens generally 2 or 3 days after the anthesis, then there was no impact on the floret fertility, obviously. So this clearly shows that 5 days of heat stress, the 2 sensitive stages are happening at the time of flowering and then few days before flowering. In case of peanut, we wanted to now have the data for detailed data for majority of the crops. Just to show you the diversity, I wanted to show you multiple crops here. The peanut on the groundnut, the temperatures are shown here. We exposed them to 1 day of heat stress, and then we were able to identify what's the most sensitive stage. Clearly, about 5 days before, 4 to 5 days before anthesis, and at the time of anthesis for groundnut. And in the case of rice, we expose them to only 6 hours of heat stress. If the 6 hours of heat stress is imposed at the time of flowering, or a few days before, 1 or 2 days before, a few hours before, then that also impacted 6 hours of heat stress at the time of flowering, or 2 or 3 days before anthesis. It negatively impacted the floret fertility. And obviously, there were some genotypic differences, which I'll talk in a little bit. So as you can see clearly that even 6 hours of heat stress or 1 day of heat stress can significantly impact the floret fertility, which will ultimately decrease the seed numbers. And here's the case of wheat. Again, the same story. At the time of flowering, 5 days before anthesis, and then this is the individual grain weight. So when you have stress, this number decreases the spikelet fertility. But if you have the stress at the time after the seed set, then it decreases the individual grain weight, primarily by decreasing the duration, grain filling duration. The two most sensitive stages for most of the grain crops is at the time of flowering and few days before flowering. So what is happening? I already explained it to you. At the time of flowering, it is the pollination, pollen tube growth, and the fertilization which is impacted. Whereas if it is happening few days before flowering, it actually impacts the viability of the gametes, whether it is pollen viability or the stigma viability. And so that's the case for wheat. And then primarily that decrease was aligns with in this case, I'm showing you just the pollen viability data, which was decreased when you had the temperature stress at 8 days before anthesis. And that's the reason why we had a lower spikelet fertility. And if you look at the drought, it also had follows the same situation. In this case, we were not able to track that effectively, that at a minute level, couple of days. But here we had the stress for more than 5 or 10 days. So again, at the time of anthesis and 10 days before anthesis, that's when it negatively impacted the grain numbers. And we believe that was primarily due to the loss of gamete viability. And then when the stress happens at the beginning of the seed filling, rapid seed filling duration, it significantly decreases the individual grain weight, because it decreases both the grain filling duration and also the rate of grain. So those are some of the impacts of the shorter duration of temperature and drought stress. Now let's look into the longer duration of the drought stress, and I'm just going to give you some temperature thresholds and response curves for major grain crops. And in this case, I'm showing you the data on the harvest index. We all know what is harvest index. Harvest index is the ratio of the grain yield to the total biomass. I'm showing you harvest index because it captures what has happened in the entire growing season. And then on your x-axis, the mean daily air temperature. It's a daily temperature, meaning the average of daytime maximum and nighttime minimum temperature. This was my early career work. So here are the temperature response curves for wheat, rice, sorghum, and millet. And there are also dry bean, peanut or groundnut, and the soybean. And then I'm showing you here the ceiling temperatures, meaning that the temperature at which there was no grain formed, therefore the harvest index was 0. As you can clearly see, there's a nice variation for ceiling temperature. Wheat has the lowest ceiling temperature at 31 degrees Celsius, rice is 35, sorghum is 38, and pearl millet is 41. And similarly, among the legumes, dry bean is 32, peanut is 39, soybean is 40. And I also looked at cowpea and mung bean, which have about 41 or 30 ceiling temperature. So we have an option of choosing the crops. There's a nice variability for different crops and different ceiling temperature. And of course, also the response curves and optimal are different. So those were the impacts of shorter duration and a longer duration of temperature and drought stress. Now I'll give you quickly, because I know a lot of you are interested in how do we screen these genotypes of germplasm collections, what are some of the important traits, and how do we measure those traits, low throughput or high throughput methodologies. So I'll just name the trait, and then later on when I talk about genetic variability, I'll come back and give you some specific examples for multiple crops. I still hope that you're able to hear me and the visuals are okay, JP?
Yes, sir. Yes, sir. Very much okay, sir. Some question is also there in chat box. So if you
Okay,
can
so
take
what
up
I'll
now,
do
or
is
you can take later, sir. Whatever it is.
let's see. Let me see. Yeah. So I'll take the questions later on, but it
Later, sir.
looks
Yeah,
like
that will be fine, sir. Okay,
this,
we can take later, sir.
yeah, I'll come back. Yes, I see the questions. So from Kirti Rani and Vijaya Jodhpur, but I'll come back to those questions.
Okay, sir. Okay, thank you, sir.
I'll follow them. So now I'll just mention the traits and then we'll go into a little bit deeper. So for heat stress or high temperature stress, these are my top 5 picks. And when I as a physiologist or as an agronomist, one of the key things is we need to make sure that whatever trait which we are screening the germplasm for, eventually that trait has to impact yield. Because if you ask farmers what are the 3 things they want, they're interested in from their crop, number 1 is yield, number 2 is yield, number 3 is also yield. So we need to be really making sure that our trait eventually, even if it is a germplasm collection later on, need to be working with the breeders to make sure that it represents and actually impacts positively the yield components. And the 3 yield components, as I mentioned to you before, is the seed plant population, seed number, seed size, and of course the biomass, so that they have a greater partitioning. So for high temperatures, first is the productive success seed set or seed numbers, and you can measure that by gamete viability or the floret fertility. And I also mentioned time of day of flowering here because that's an important trait. Because if you look at daily, the temperatures will be cooler in the morning, will go up from morning to this noon, and then solar noon, it reaches maximum, and then later on the temperatures come down. So if you have a genotype or a crop, which is actually fertilizing, pollination and fertilization is happening at a cooler part of the day. Meaning if as soon as the sun rises, the pollination is happening and from pollination to fertilization, it's about 30 minutes to an hour or 4 hours maximum, depending upon crop species. But generally it happens very quickly. So once the embryo is formed, pollination and the pollen tube growth and fertilization is happening in the cooler part of the day. Once the embryo is formed, embryo is relatively tolerant to these stresses. So it'll form an embryo and it'll stay put there and it'll wait for the environmental conditions to come back. So that's what I mean by time of day of flowering. And it has been explored well in rice. And I'll give you an example later on. But and if you look at some of the crops like cowpeas and pearl millet, actually the pollination and fertilization happens very early in the morning or even some cases during the night time. So that's probably the reason why those crops are much more tolerant. And the second one is a favorable respiration. We've spent a lot of bit of time or effort on photosynthesis, but not much has been done on the respiration rates. So but we all know that the net photosynthesis is the difference between the gross photosynthesis and the respiration. So we only have looked at half of that. We're focusing on photosynthesis, but we also need to be looking at respiration if we can find optimal respiration so that the net carbon available is higher. And of course, grain filling duration and grain filling rates, if those looking for those which have a longer grain filling duration and the higher grain filling rate, that will impact the seed size will increase. And increase membrane stability, because one of the first things is temperature stress and drought stress or the nutrient stress or salt stress would do is it damages the membrane. So those genotypes which can maintain higher membrane stability will be good. And also canopy temperature depression is another trait which has been exploited very well in wheat. So meaning that those genotypes which are able to keep the canopies cooler are actually avoiding the stress. But that comes at the cost of water, because to keep the canopy cooler, they have to transpire, and then there needs to be irrigation facilities, so water should be available for that trait to express. And both of them will give you longer green leaf area duration and also higher biomass. Therefore, the partitioning will be better. So those are my top 5 picks for the for the heat stress. And now looking into the drought stress, one of the most important trait which has been exploited very well is the stay green or the longer green leaf area duration, which has a slower senescence so that there is photosynthesis available during the grain filling period. And the second trait, which has also been exploited very well, but recently, is called limited transpiration trait. So I'll show you a specific example. What this trait does is that it saves the water during the early stages of the crop, and then make that available at the later stages of the crop. And we all know that later stages of the crop, or the reproductive phases, are much more sensitive to water stress compared to those of the vegetative phases. So limited transpiration trait is one of the things which have been recently, about 10, 15 years ago, they have been identified, and the genotypes in those traits are coming up now. And we all know about the value of the water use efficiency or the water productivity. And then efficient root systems, whether you need a root angle or the rooting depth, whether you need a narrow roots or you need a deeper roots, wider roots, that depends upon the rainfall or the environmental conditions. If you are dependent upon the intermediate rainfall, if you are dependent upon the rainfall, you are looking at a wider root, and then those will exploit larger areas. But if you are dependent upon the water from the groundwater up, then you are looking at a narrow roots and the deeper roots. So each of them has a different value. And also canopy temperature depression here, because as I said to you before, canopy temperature depression comes at the cost of the water use. So you're looking at those phenotypes which can maintain the higher leaf temperature, not wasting the water, but of course making sure that they also have a higher yields under those conditions. And we were able to identify some of those type of cultivars in multiple crop species. So those are my important traits for temperature and drought stress. Now let's look into how do you measure this? What are some of the physiological measures, low throughput, medium throughput, and high throughput methodologies for measuring these traits? Directly, those traits are the surrogate traits, which ultimately give you those impact on those physiological. So I've summarized here in these 8 items here. Again, dividing them through vegetative stages and the reproductive stages. So some of the tools, some of the measurements you can do is measure cell membrane thermostability. You can measure photosynthesis or stomatal conductance, canopy temperature, and the root traits, and that can be done throughout the crop growth, but generally it is much more valuable during the vegetative stages, and then especially the root ones, and it indirectly gives you the impact on the resource use, the leaf area, and the biomass. So if you're looking at the reproductive stages, which may have impact on seed numbers, seed size, of the partitioning, you're looking at the pollen, stigma, viability, or the embryo abortion, or the viability of the embryo, and the seed size percentage, and stay green rate of the increased green leaf area duration, and improved harvest index of the partitioning. So these are the broader traits which are valuable for both heat and drought tolerance. And then I'll now show you some of the measurement tools. First, we'll talk about low throughput measurement tools. So this is for photosynthesis. You have typical IRGA, and here I'm showing you the Leica portable photosynthesis machines and the CID ink ones. This one is a little bulky, and then this is a little bit portable, and you can use this at any stages of the crop growth, pre-flowering, flowering, and post-flowering. And then these IRGAs will give you photosynthesis, tomato conductance, transpiration, and also leaf temperature. But of course, it's very time consuming. Each measurement might take you anywhere from 10 minutes or higher, depending upon how quickly you can calibrate and how well your conditions are in microclimatic conditions and the conditions within the chamber are. So it's portable machine. It gives you direct measures of those, but it is definitely not a high throughput methodology. And then the second one is a chlorophyll fluorescence. You can use a pulse modulated fluorometer, and nowadays you can also get a fluoropen, and this will indirectly give you the membrane stability or the damage to the membranes. And this is reasonably high throughput, but of course sometimes you might need to adapt. You can measure either dark adapted or the light adapted one, so you have to put the clips on the leaves and then use this instrumentation after the for the dark and once the adaptation to the dark has happened. And if you can measure either F0/fm or FV/fm, which indirectly gives you the damage to the fluorescence and then damage to the thylakoid membranes or the membranes. So reasonably fast, but you have to dark adapt it if you want to get the 0/. And then to measure the green leaf or greenness of the leaves, handheld chlorophyll meters are popular. And then this is very quick. You can just clip onto the leaf, and then it gives you a chlorophyll index, which is an index which is positively correlated with the chlorophyll content or chlorophyll concentration. So that's pretty rapid. So you can see if some of the leaves are senescing rapidly. And if you have the time series data on that, you can actually quantify how quickly the leaf is senescing. If you have a daily measures or alternate days at the same point of the leaf, then you can actually measure the rate of senescence. You can capture the stay green trait for this. And then the next one is the canopy temperature. And in the past, we used to use the pointed temperature guns, but we are not using that anymore. It suggests it gives you a single point measurements, and there is a lot of variability. So nowadays, we are using more of a thermal imaging camera that actually it takes the image of the entire leaf, and then it tells you what's the temperature of that leaf is. And it is one-time measurements. But now you can also fix these sensors or you can also put the thermal camera or even the radiometer, simple radiometric measurements of the temperature. And that way they can continuously monitor the entire canopy temperature. And then it gives you how cool or how warm the temperatures are, leaf temperatures are. So these are relatively high throughput, and these are automated nowadays. You can continuously monitor. And then when it comes to the reproductive traits, pollen assays measuring the pollen viability, whether it is through staining technique, in vivo or in vitro. And in vitro, you can measure you can collect the pollen and then put it in a germination media, which has to be standardized for each of the crop species. So generally, it is agar medium plus some of the micronutrients which are mixed in there. You can also do semi-solid medias. So you collect the pollen, put it on them, and then measure the pollen tube growth, whether it is germinating or not. Then you can also have the light stains on the viable stains, which will only tell you whether the pollen is viable or not. It's not necessary that all the viable pollens are going to germinate. So this is not easy. It's time consuming. You have to collect the pollen very early in the morning as soon as anthesis happens, because the viability of the pollen grains loses very quickly once the anthers are dehisced and the pollen is not connected to the mother cell, and the viability is lost pretty quickly. So you have to do it right when the flower opens. It's a time consuming method as well. And this is just to give you an example of once it's in the agar medium, you also have to measure it very quickly so that you can actually differentiate between the germinated and the non-germinated pollen. So there are opportunities in any means. It is not it takes a lot of time. And nowadays we are trying to see if we can use artificial intelligence to determine to take an image and then use that to artificial intelligence and see if there is a way to not manually count it, whether the machines can count the number of germinated and non-germinated pollens, and then you can measure you can get a percentage of viability. So those are some of the traits for heat tolerance and drought tolerance as well. But when you look at drought specifically, there's two components for drought: root angle, and then the rooting time. And then root angle meaning that you can measure them. And this is just one of the techniques which was published. So you can grow the plants in these pouches, and then you can put them on the agar media or in the pouches to look at what the angle of the roots are, whether it is a wide root angle or you have a narrow root angle. Generally, if you have higher root angles, they're shorter depth. Then if you have a narrow root angles, then they are much more deeper. And generally, it's good to measure just the early vegetative stages, and that angle is pretty much fixed during that time. So that's also very time consuming as well. But and then if you're looking at a different when you're looking, if you want to look at in the real conditions in the potted plants, these are tall, and these are 30 centimeters or 20 centimeter diameter, and then you have one. So you can grow in an artificial media called turbo, and then you can plant them, and you can actually extract the entire root. And then once you have these roots, you can pass through the machine and actually measure the total number of roots, the fine roots, and then what is also the root length density. So anyway, it's not a it's not a high throughput. It takes a lot of time. So those are some of the traits or the tools which you can use for measuring some of the physiological traits. Then what about the high throughput techniques? Because obviously those are good if you have a known susceptible and known tolerant. If you're looking into the mechanisms of susceptibility or mechanisms of tolerance, then those tools are valuable. But if you are a breeder and if you want to screen a large germplasm collections, none of those machines, except for the fluorescence and the canopy temperature and the span meter, are still medium throughput, but not high throughput. But now there is a lot of progress which has happened in terms of our ability to use remote sensing tools, whether you're using a handheld or a spectroradiometer or a thermal imaging camera on the ground, or you're using UAVs and also fixed wings, or even go all the way up to the space. So you can measure these. We have significant advances in terms of sensors, which can be mounted on various type of machines to be able to measure some of those traits. So we most commonly use the drones, hexacopters or a fixed wing type. And then now you can put those cameras on those and then allow them to put them and then take the images. And now they are very sophisticated. You can actually set the path and then mount those cameras, whether it is spectral camera, digital camera, or thermal camera, and set it up. Those will go around those flight path which is set up, and then give you the images. Of course, one of the conditions needs to be that it's a cloud-free day, so that your data is not confounded by other traits. So here are just some simple examples. There's a winter wheat association panel, which was laid under irrigated and dryland conditions. These are, you can see individual plots. You can look at the reflectance, multispectral data. That way, it tells you which of those plots is drier or which of the plots is impacted greenly, which of those plots or varieties are impacted by drought stress, especially if the greenness of the leaf is lost. So, and if you fly it low enough, you can actually even distinguish between the plants, and then not only the plots, but also you can look at the individual plants as well. And that was a spectral camera based on the reflectance, kind of gives you the greenness of the leaf. And you can also mount the thermal imaging cameras on those, and then you can actually measure the temperatures of the canopy itself. There's a wheat temperature map, canola temperature map, and the sorghum temperature map. So nowadays you can which gives you indirectly the canopy temperature, which indirectly gives you this matter conductance of the transpiration ability. So you have this high throughput phenotyping tools now available. But it's one thing to take an image, but you have to make sure you understand that image and how do you interpret the data and then use it in your evaluation is a different ball game. That's where most of the emphasis is happening now. So those were some of the tools, and now we'll look into what options do you have in terms of management. Excuse me. So we'll look into what type of, how do you manage the stress? Basically, we have two options. One is the agronomic management. Another one is the breeding approaches. So I'm just looking at the time. I'm at 44-minute mark, so I'll be okay. In terms of stress management, first let's talk into more of an agronomic and the crop management practices. You have multiple options here. For example, depending upon the environment which you are, and if you know when the stress happens, you have multiple options. But generally, from agronomic perspective, early planting, maintaining the plant population, and increase so that you have a higher radiation use efficiency, higher light interception. And then if you have irrigation option, make sure the plants are not in stress, and also balance nutrients in terms of NPK and micronutrients, and then also using the plant protection so the plants are not stressed. So that's one option. And there's been a significant use of hormones, growth regulators, antioxidants, osmolytes, and more recently, nanoparticles and microbials to manage the different levels of stress. I'm not going to go into the details of those, but we have multiple options in terms of spraying the plants or even seed treatment of the plants to make sure that they are relatively more or tolerate some of the stress when it comes. And from a varietal selection, you have a true tolerance, you have either escape, you have the true tolerance. So depending upon again environment and what type of stresses and when they occur, you may choose either short duration or longer duration. You can also choose the plant architecture. And one of the significant progress in terms of architecture has been the leaf angle, the narrow leaf angles, so that you can increase the plant population and there's a more light interception happening. Early flowering and early morning flowering, I talked about that. Gamete viability, I showed you that as well. And if you have a higher gametes or pollen production, and of course photosynthesis, leaves and spikes nowadays as well, especially in the flag leaf and also the spikes of the wheat. There's significant research done on the ability of those spikes in their green awns and their value in the providing photosynthesis to the grain roots and water use and harvest index. And then in terms of tolerance, you have pollen tolerance, stigma tolerance, cyclic fertility, rapid grain filling, and also the longer grain filling duration. And I mentioned about canopy temperature and stay green trait. So those are some of the ways you can manage the stress in terms of selection of agronomic and then also varieties and the tolerant genotypes. And of course, there's I'm sure you have heard from many of the scholars here about various breeding approaches to address some of these stress tolerance. I'm no expert in this area, but I'm sure many in your course, there are a lot of people who probably talked about each one of these various approaches in terms of plant breeding for stress tolerance. I'll quickly just provide an overview. Conventional breeding, one of the most important thing is exploring the genetic resources within the species, but also close relatives or the wild relatives of those species, because that's where most of the tolerance is. Then once identifying the tolerant or susceptible lines, and then using those lines into your regular breeding program to control breeding, pedigree breeding, population breeding. Now you have options of fast breeding as well. So there are multiple approaches that you can do there, and then and there's the technologies like double half-life, which has really enhanced the breeding cycle, including the fast breeding, speed breeding, so to say. So those approaches are there. And molecular breeding using mapping the genes, QDLs of interest, linkages and association mapping, marker-assisted mapping, marker-assisted recurrent selection, genome-wide selection. So there's a lot of opportunities there as well. And of course, you all know about the transgenic approaches, candidate genes, functional characterization of those genes, and then and genetic transformation. And more recently, we have gene editing tools like CRISPR-Cas9 for target mutagenesis for accuracy, and CRISPR-Cas12 for novel gene editing tool as well. And there's been a lot of progress in the recent last 2 or 3 years in terms of using those for stress tolerance. And I don't need to tell this group that this is This is the non-GMO methods, especially the CRISPR-Cas9 technology, whereas the other approaches are transgenics, and there are some restrictions on using transgenics, particularly when it comes to India. So those were some of the approaches. Now I'll go a little bit deeper into non-generic resources and what type of opportunities do we have, and then what are some of the research done in core crops. I'll give you examples from sorghum, wheat, rice, and then I believe soybean. Those four, as I mentioned. So this is very particularly appropriate for NBPGR and this particular group. And Jyoti is a leader in that area, so I think I'm sure you would have heard from her or others who might have spoken about that. So plant generic resources. This is one of the most important component for exploration for tolerance and resistance. And this is where most of those sources are. And once you identify those sources, then you can use them in your breeding program. And there's a lot of generic resources. I'm just giving you some examples. Just in NBPGR, you have. I just picked up the most important crops: rice, wheat, maize, sorghum, pearl millet from cereals, and you have a lot of collections there. And I'm not going to read you these numbers. There's huge collections there, and just in NBPGR and India. And for legume crops, chickpea, pigeon pea, mung bean, anywhere from 14,000, 11,000, 4,000. And then oilseeds, groundnut, brassica, and sesame. So these are just what you have in India. And then I'm also showing you some examples of the CGIAR organizations or others. And you seem to have a pretty good collections right there in NBPGR. But if you look at CG centers, rice is 132,000 at Erie, wheat is 150,000 at Sumet, maize is 28,000, Sumet, sorghum is close to 42,700, or 800 acres, pearl millet 24,600 and change. For legume, chickpea, also acresat, pigeon pea, acresat, and then mung bean. Sorry, was spelling mistake there. Mung bean, that's World Vegetable Center or AVRDC. And groundnut again, acresat, brassica, and sesame more are global. And China and India probably has a very good collection of sesame. We have a large, very good collections in these gene banks. But of course, it's very difficult to screen all of these big germplasm collections. So one of the approaches which has been very popularly done is capture that genetic variability, which is there in the larger germplasm in the smaller collections. And that's where the concept of core collections and the mini core collections comes into picture. And I'm just giving you the mini core collections, meaning that whatever that genetic variability for sorghum was there in 42,700, that was captured using various approaches to make sure that variability is captured in a fewer number of genotypes or accessions. And those could be can be screened effectively. And this methodology was popularly done by Hari Upadhyay, and Jyoti was telling that he was here to talk to you, or he was here, or he's going to talk to you about his approaches. So he has developed these mini core collections of sorghum, pearl millet, groundnut, chickpea, and pigeon pea. And I'm sure he must have done it for other crops as well. But I'm just giving you these four crops, and these range from anywhere from 240, 30 to 148. So these are all at the Icrosat gene bank, which was named after Aris Baroda because of his contribution and his vision of having these collections at Icrosat. And I'm sure I know that he either spoke to you or will be speaking to you very soon. So it's again. So now once we have these mini core collections, then these could be reasonably screened to look at those various traits which I just showed you. So now in the next 10 minutes, I'm going to wrap up, and then I'll give you some specific examples of how I have utilized or how other people have utilized. I'm just mostly going to show you the data from our lab, how we have used some of those mini core collections or some of those other collections to identify some of the stress tolerant varieties, and then later on pass it on to breeders, or we do some pre-breeding work, and then they were used by breeders to incorporate some of those traits into elite lines. So I'll start off with rice, and then we'll go to sorghum, and then to soybeans. I'll show you those. So first, I would like to start with this trait called early morning flowering. So and originally I started work on this in 2005 or 4 or 5, and that's when we had a project from IRI. So what we did was we looked at different species of rice, or Rhiza sativa, or Rhiza glaberrima, and some interspecific lines to look at what was the time of their flowering, meaning when they were flowering. So you can see the interspecifics of the glaberrimas were flowering very early in the morning. They were flowering at 7 a.m. or even before, and the pollination was happening that time, and the fertilization was completed in the next 30 minutes. Whereas the Rhiza sativa ones were ranging from later part of the day, 9, and even some went up to about 12:30 p.m. So that shows you that those lines, most of the Rhiza sativa were flowering very late in the morning or in the afternoon, and that's when the plant was being exposed to very high temperatures. So what it was impacting was the reception of the pollen on the stigma, pollen germination, pollen tube growth, and the fertilization. So interspecifics were very valuable, and later on, they developed the nil lines for this neoisogenic lines, and were able to look into the mechanisms, and then that trait is now being utilized in the rice breeding program. So another one that was early morning flowering, and then seed set percentage. There's huge genetic variability there. I'm just showing you a few examples here. And then N22, Nagina 22 was one of the most tolerant one, where the pollen production was not impacted by the high temperature stress. Not only that, but the reception of the pollen on the stigma was also very high compared to the susceptible lines, where the pollen production, this is the percentage of the ambient temperatures, so very little pollen production under high temperature, and also the reception of the pollen was very low on the stigma. So therefore, the flatlet fertility was negatively impacted, or very low on those. So this of course need a lot of effort and work in terms of pollen collection, and then measuring the reception of the pollen itself. And then another trait is the favorable respiration, which I mentioned to you. And this work was done by Dr. Krishna Jagdish at IRI, and then later on he moved to Kansas State University. And then here he screened especially the rice genotypes for higher nighttime temperatures. And here is the relationship between the respiration and the grain yield. The higher the respiration, the lower the grain yield, and then lower the respiration, higher the grain yield. And these are some different genotypes. So there was genetic variability in terms of respiration, and then generally the lower or the optimal respiration plants are the better ones, because the net carbohydrates available in those plants to go into the yield was relatively higher than those which have higher respiration, and therefore the lower grain. Again, this is not high throughput, but it takes significant amount of time. So that was on the rice. And now let's quickly look into the wheat. And here is the wheat genotypes, which was Carl 92 and Wettner. So those are the two different genotypes. Wettner is relatively more heat tolerant compared to Carl 92. And here we looked into the optimal temperatures, high nighttime temperatures, high daytime temperatures, and combination of both high day and nighttime temperatures. And I'm showing you the various data on yield components, grain numbers, individual grain weight, grain yield, and then grain yield per plant and harvest index. So there was clear variability in terms of Wettner was much more heat tolerant to both the high nighttime temperature, high daytime temperature, or combination of those two. And in general, the combination of those two, high day and night, was much more damaging compared to that individual stress levels. Then later on, we screened even a wider germplasm collection. This was the work done by, in collaboration with Washington State University. They had the collections of spring wheat and collections, which were screened for heat tolerance, both at the time of flowering and the grain filling stage. Here you have the data on the seed numbers, seed weight, and the harvest index. Again, quite a bit of variability in the seed numbers. Some of them were extremely high as 50 spikes, seeds per spike, and then and then some were very less. The same was also true for seed weight and harvest index data. So there is quite a bit of genetic variability. We used those, and then we were able to categorize them based on the various timing of the stress, whether they were sensitive at flowering or whether they were sensitive at grain filling stage. And then we categorize them into resilient, sensitive, and highly resilient genotypes. So there is quite a bit of genetic variability. And we later on went ahead and looked into the pollen germination piece of that as well to identify some of the tolerant lines, which are now being used for breeding for heat tolerance. And then we also looked at the wild relatives of the wheat. And here, there are different agelubs, Spaltoides, geniculata, McGrathie, Cersei, and Longishima. So and compared to that Sativus, compared to that Triticum east one, there's a quite genetic variability of this there. So we screened, one of our students screened a lot of these accessions and looked into those, and some of those species had differential response to high temperature in terms of various yield traits. We were more interested to look at the relative performance rather than the actual, because these are wild relatives and not elite lines. So that was and we identified some of the lines within each of those species which were more tolerant to high. And now same thing with grain sorghum. And here we again I screened most of the mini core collections, entire mini core collection of sorghum for drought and heat tolerance. And here's just to show you some of the variability for pollen viability. We're using staining technique and also in vitro pollen germination. There was impact of high temperature stress on that, and we were able to identify some of the lines. And one of the things which we did was look at the cardinal temperatures for pollen germination, meaning that we collected the pollen and then developed this temperature response curve by incubating the pollen at different temperatures. So you get the base temperature, you get the optimal temperature, and you get the sealing temperature. And we looked at the sealing temperature and then categorized them into whether they were tolerant or susceptible, potentially meaning that they might be able to perform better when they were exposed to higher temperatures at the time of antithesis. So we looked, the range was pretty high, anywhere from, and some of them were 45 degrees Celsius sealing, some had 40. I'm just showing you some variability here. And even the optimal temperatures were slightly different. And the pollen germination percentage itself was also very. So there was that genetic variability, which could have been exploited for that as well. And then the fourth crop, which I'm going to show you, is soybean for heat stress, where we looked at PI lines, plant introduction lines, and also elite germplasm to look at the impact on pod numbers and seed yields as well. And then again, the same thing we did for pollen, cardinal temperatures for pollen, and then looked at if there was any variability for the cardinal temperatures, specifically looking at the sealing temperatures for high temperatures. And we are continuing this work, and then our breeders are utilizing some of these lines and then particularly putting that trait into the elite line. So that was about the high temperature stress. So now I'll wrap up with just giving you a few examples for drought stress. Again, starting with wheat, we screened the entire winter wheat collection and the spring wheat collection, about 300 lines, and then looked into the impact on the seed numbers here, and then determined which one of them were impacted more. And then we did this under dry land and irrigated conditions as well, and categorized them into drought tolerance or drought susceptible. And then we also looked at the entire root traits and root angle and root length. And here's an average root angle. Some of them had lower root angle, some of them had extremely high root angle. In the root length, we didn't find too much variability in this particular collections, but that could also be utilized as one of the potential traits. And then sorghum, we had the diversity panel, which consisted of 300 lines, and we used the thermal imaging camera, and then the canopy temperatures, and also the greenness of it's using SPAD and also the spectral radiometer to look at those temperature, the differences. I'm just showing you the leaf temperature data here. So we were able to identify huge differences. Some of those were keeping cooler canopy, some of them were keeping warmer canopy, and there was a huge variation in the grain yield. We were interested in this particular square where there was higher leaf temperature and then higher yield. So that's a true tolerance. And those who were keeping the canopy cooler and then were higher yield, they were avoiding the heat stress. And that comes at the cost of water. So these have potential value in terms of true tolerance. And we went ahead and looked at the genetic, what was the reasons, and also I mentioned to you about the limited transpiration traits. So there we screened those limited transpiration rate at different vapor pressure deficits. So there were some genotypes which continues to increase the transpiration with higher vapor pressure deficit, and some of them had higher linear and then they had a break point, and then some had more linear and then had a break point. Each of these will have a different in different environments they'll be value, meaning that the ones which were lower rate and then early break point, they were using less water, and that water will be available during the grain filling point. So that those traits now this limited transpiration trait has been observed in majority of the crops. First identified in soybeans, then in sorghum, then in groundnuts, and I believe it's also available in wheat now. So that's another trait. And rooting depth and root angle, very hard. Use the turf-based media, grow the plants, harvest, and then scan these roots in terms of depth, number of fine roots, number of different diameter root, and nutrient density. Very low throughput and a lot of time consuming. And for drought stress in soybean, we did the same thing. Grow them under irrigated and dryland conditions. Wide genetic variability for seed yield, transpiration rates, and then also photosynthesis as well. So that summarizes. So I took almost one hour, 5 minutes. So we would like to conclude saying that abiotic stresses like high temperature and drought can cause significant yield losses. Short episodes, even for a few hours or a few days, if they occur at critical stages of crop development, can negatively influence the yield. And particularly 2 stages are very sensitive: gametogenesis, which happens a few days before anthesis, and at the time when the viability of the pollen grains is being determined, and at the time of flowering, when pollination, pollen tube growth, and fertilization happens, and sometimes even early grain filling. And crops vary in their response to stress, have different thresholds, and genotypes within the crop, within the species also vary. And we need to systematically screen larger germplasm collections, especially starting with mini core collections and also going to the wild species to identify the tolerance. And then there are several physiological, biochemical, and molecular traits which confer the stress tolerance. And we can make, once if you are using any of these, we need to make sure that functionality of these genes are expressed at a plant level or at a crop level, and it has a positive influence on the yield. There are medium and high throughput phenotyping methods, particularly the remote sensing techniques with use of spectral radar, spectral cameras, digital cameras, and thermal cameras will allow you to screen the larger germplasm collections. And once those identified tolerant lines are there from your plant genetic resources, they must be used in a targeted breeding program. And not only the breeding, but also we have options of agronomic methods to manage the stress efficiently. With that, I would like to thank the various teams. I particularly chose this visual because maybe Jyoti will identify a lot of people there. So that was almost 10 years now. I think here you are, Jyoti, and you probably
Yes, sir. Yeah, I can recognize very well.
and
Yeah.
you've grown, I've grown as well. And here's Tara as well. So
Yeah.
these are the entities.
Dr.
And
Tara, Dr. Aarti, Shruti.
yeah, so Aarti Bhatia was there too.
Yeah. Sanjana, I think.
Sanjana Reddy was also there. So yeah, and then I spent these were the 2 people. I think before you, Sandeep was also there. Both of you are there in yeah. For that, I really wanted to thank you for giving me this opportunity to speak to you today. But I did not have a chance to talk to you about my Sustainable Intensification Innovation Lab, which I've been leading since 2014. If you would like to learn more, please visit us on the website and follow us. I'm not going to go through that, but it's a great. This is my current team, and we are celebrating the 75th independence, and then this year India takes the lead on the G20, and also has been declared as a year of millets 2023. And we have a tremendous opportunity to show the world where India stands and how it can contribute not only for food and nutrition security in India, but also what it can provide to the rest of the world. With that, thank you very much for giving me this opportunity. And if you have a question, please unmute yourself and
Okay, sir.
ask the question.
So
Or
we'll
if you-
take, sir, a few quick questions. So in chat box, one question was there.
Mhm.
Does CTT affect overall development of sorghum, and can it be used as selection criteria for maturity?
So
Welcome, sir. Dr. Viswanathan, sir. I welcome you.
Yeah, thank you.
Yeah, just we started a little
Yeah,
late, sir.
I just missed seeing Professor Vora. Just I joined. I was not knowing.
yeah, that's fine. That's fine. I think yeah, we are
Thank you,
team
Professor.
tagging here. Yeah.
Thank you so much for your time.
Yeah, so we started a little late, sir. So just
No, please.
few minutes.
Continue.
Okay,
Yeah,
sir.
continue. Please continue. It is a rare
Yeah.
opportunity for the participant to interact with
Mhm.
Professor Vora. Please continue.
Okay, thank you, sir. Okay.
So yeah, can you ask a question, Kirti?
Yes,
If
sir.
you're here, it maybe just better done rather than.
Does
Um,
canopy temperature affect overall development of sorghum, and can it be used as selection criteria for maturity? By
So,
Kirti Rani.
is it? I mean, does she really mean canopy temperature or CDT?
Kirti, you can ask.
Hello, am I audible, ma'am?
Yes, you're audible.
So actually CTT here is cumulative thermal time accumulation. I wanted to know if it has impact on different type of plants with different phenological states, like day neutral plants or short day plants or the long day plants. How
Yes,
CTT
absolutely.
can be used in these type of plants,
Yes.
different categories of phenological states.
So yes, growing degradase concept is commonly used. Growing degradase, and that's very commonly used, and it can be used in sorghum and maize or any other crop as well. But one thing which you have to be careful is that if there is a maximum temperature set there, meaning that especially those phenological events will be actually decreased, will not happen at a higher temperatures. So you have to set, when you're measuring that growing degradase, you have to set a maximum temperature beyond which you don't accumulate any more thermal degradase.
Okay, so if a participant may ask if they have any further query, or they can ask later also to sir through a mail.
This is the time I'm here, so you might as well ask now if you have.
Yeah, please ask now.
Don't be hesitant.
Yes, please.
Sir, here I want to add that apart from the CTT, like cumulative thermal temperature accumulated by the crop for the whole development stage, plus humidity, if it will be taken into account, then how it can be taken into one equation. Both
Usually,
humidity in the soil, humidity in the plants, and then thermal temperature accumulated by the plants. All three can be accumulated to one equation so that this can be used as a criteria for the early maturity.
generally people are not using the humidity levels, but generally it's done by the growing degradase thermal times. It's just the temperature. Unfortunately, humidity is not being regularly used. But sorghum, I think it's not an issue. But temperature is a primary driver. And
Sir, another question is there, sir, from
yeah, I'm reading it.
Okay, sir. Okay.
In the rice, rice is generally, depending upon which type of rice, if it is flooded rice or aerobic rice or
Aerobic rice.
Aerobic rights, yes. Aerobic rights, I think then I would say that it's not the depth, but it's the total route length would be valuable. And more importantly, the route length density probably is a better criteria for route harvest. I would look at route length density, not the or the number of fine routes as well.
Yes, sir.
Okay. Yeah, anybody want to ask? Okay, if not, then means they can further send in chat box. So we'll send to Dr. Bara, or you can directly mail to Dr. Bara for any clarification of dot.
Yeah, sure. And I'll just type my email address so you can. In the
We'll
chat
be
box.
sharing to them, sir. We'll be sharing to
Okay.
them the email ID also. Okay, so now I would like to thank Dr. Bara for his comprehensive talk and giving detailed accounts of abiotic stress and crop yields, their impacts, evaluation and management. So, sir, you have covered in detailed way the effect of short-term stress, long-term stress, all phenotypic methods and tools, and then stress management approaches for PGR evaluation. So certainly our participant might have enjoyed this knowledgeful lecture, sir. So, and we are also very grateful to you, sir, your greater role in the support and strengthening human resource development, particularly for India, sir. So you are training and mentoring so many students and scientists and faculties from the India. So thank you very much, sir. And I hope that our participants, they may also, if they get the chance, so they can go to SUS lab and learn all the techniques and tools and knowledge. So thank you very much, sir, for your time. And you are awakening until late. I think it is now 12, sir. There.
Almost, yeah.
Okay. So thank you very much, sir.
Yeah. And appreciate the opportunity and very good to see you, Dr. Vishwanathan. Yes.
Thank you, Professor.
Yeah, and then convey my regards to everyone. I think Jagdish Rane is also going to be speaking in the same session. So, but I won't be able to hear you.
No.
Sorry.
Later.
Sorry.
Better hear this earlier.
Alright.
Thank you so much for giving lecture at this point of time for all the participants.
Anytime.
Okay. Thank you, sir.
Bye.
Okay. So, dear participants, so now we have our next speaker. So, before sir will start lecture, I will just give a quick introduction about this, sir. So I think the slide is visible. So today's eminent speaker for the second lecture is Dr. Chinuswamy Viswanathan. So sir is at present Joint Director Research, ICR, Indian Agricultural Research Institute. Sir has completed his graduation from Agriculture College and Research Institute, TNAU, Kilikulam. And after that, he completed his master's and PhD from IARI, New Delhi. Then sir joined ICAR in 1996, and he became served as a senior scientist and then principal scientist since 2010 onwards. And sir also headed division of plant physiology from 2013 to 2022. And as now sir is JDR. Before that, he also served as acting JDR. So sir, the research area mainly included plant physiology, molecular physiology, phenomics, and now sir is doing a greater job for genome editing also. Sir has received several awards and honors like ICAR Jawaharlal Nehru Award in 2001, DST Boys Cast Fellowship Award in 2001. Then sir is Fellow of Indian Society for Plant Physiology. He has also received Indian Society of Plant Physiology J.J. Chinoy Gold Medal. Then he has visited several universities abroad. For example, he has visited University of California during 2008 to 10, Then he has visited Department of Molecular Biology, Harvard Medical School, Harvard University, Boston in 2009, and several other countries he has visited. Regarding publications, sir has published more than 250 research papers and several book chapters, technical bulletins, and edited several books. Sir has established the phenomics facility of the international class, and which is very much being utilized for the screening of all the material germplasm as well as mapping population. Regarding citation, so he has a great citation, 18,532. So sir, we welcome you all to the NBPGR in this international training, and for accepting our request to deliver the talk to the participant. Now I'm, without wasting my time, I'm stopping my slides, sir. And sir, please start your presentation.
Yeah, thank you, Jyoti. Am I audible?
Yes, sir. Yes, sir.
Is clear? Voice is okay?
Yes, sir. Voice is okay, sir. Okay, sir.
So thank you, Dr. Jyoti, for the kind words of introduction. So it is my great pleasure to interact with all the training participants. So they are all from ICR Institute or from?
Sir, quickly, I don't know, sir. Most of them are, sir, from SAUs, means
Okay.
about
ICR and
50 around, from ICR 9, and then from CG Center 2, some private institute 6, like
Okay.
that. They are mainly from, sir, different breeding backgrounds,
Okay,
working on different crops, sir.
okay.
That.
So that is fine. So then since everybody is from agriculture background, I need not give an introduction about the institute, the Indian Agriculture Research Institute. Everyone must knows about it. So I am from the Indian Agriculture Institute, currently working as Joint Director Research. Otherwise, my specialization is plant physiology, and as Jyoti told, I am working on a diverse aspect, and plant phenomics is one of the aspects which I started a few years back into the multidisciplinary new science. And so today I'll be, my purpose of this lecture is to give an introduction to phenomics. So the idea is I will say about a different sensor and what purpose it can be used, so that not necessarily like you should have a facility like what we have, because those kind of facility may not be available in all places. But once you understand the principle and you can apply it in with small low cost facility, also you can develop and apply it in different places. So I'll be talking to you about the various aspect of plant phenomics and introduction to it, and then I will also give some results from our work, what can be done with this kind of facility. So this one, I think all of you might have seen this picture and listened to it from several speakers in almost in all the conference which is related to agriculture. So the major question everybody is asking is how to feed the 10 billion population by 2050 and without using more land and more resources, while reducing the global warming effect, that is methane emission. And enhancement of, so the answer lies, there are several approaches to achieve this. One of the aspect is to enhance the productivity of the crop per unit input, unit time, and unit area which is necessary, and where the germplasm resources are the key. Through breeding approaches, you can improve the per day productivity, per unit input productivity, so that you are able to achieve the goal. So currently to achieve the food, feed, and fuel demand by 2050, various estimates is we have to increase the food production from 50 to 70% within about 20 years. So for this, the current rate of genetic gain is only about 1% or so. So it is expected that we should achieve a genetic gain of 2.5 4%, so that we will be able to meet the demand for various needs. So it is not only the food purpose, but also for the fuel purpose. For example, now in India, by 2025, we have to mix about 20% bioethanol with the fuel that we are using, which means we need almost about 17 million tons of food grain diverted only for that purpose, because only the first generation biofuel production is cost effective at present. Second generation biofuel production is not so cost effective, and it is very difficult to achieve the target. So we need to increase the productivity. The challenges which water scarcity is increasing, and the soil health is affected. The climate change is also another challenge. So you have to address all these aspects, and the germplasm resources are the key. And already you are in the institute who is custodian of the germplasm in India. So I think already you might have been exposed to about the importance. So when we talk of genetic gain, as I told, we need to increase from 1% to say 2.4%. Since most of you are from the breeding background, I need not explain this equation. So the change in genetic gain, it depends upon various component, the selection intensity, selection accuracy, genetic variation, and generation time, and also the inbreeding depression, or when we assemble different genes from different germplasm, unwanted genes and alleles also get. So that is negative effect. So in this, the phenotyping play a very important role. One is, as I told, it is the selection accuracy. It is very important, and precision phenotyping by using phenomic approaches can increase the selection accuracy, and also the time that we devote. Like for certain phenotyping, we need to conduct multiple years of experiment. Like for example, if we are doing phenotyping for drought stress under field condition, it is highly uncertain whether you will get drought or not, and at what stage you will get. There are so many issues, so you have to do multiple years of testing. Similarly, for temperature, or if you want to measure water use efficiency in the field condition, it is not possible. That's why not many genes have been mapped and fine map for water use efficiency in the crops. Only Arabidopsis gene is mapped. So some of these difficult traits can be addressed by using the precision phenotyping and phenomics approaches. So again, this is for general leader, and since all of you are from the biology background and breeding background, we know this, that is the phenotype is interaction of genotype into environment. And the environment, when we do it in the uncontrolled condition, the environment effect is more. So it is very difficult to measure the traits heritability more accurately. And then we need multiple years of experiment. So that's why we use various controlled environment approach as well as precision phenotyping approach, where the manual errors are less. So the precision phenotyping, or rather in general phenotyping, is key for establishment of phenotype genotype relationship. This is very important for crop improvement as well as for the crop management also nowadays. This is very much useful. That is the sensor-based phenomics approaches are very useful in the precision farming, in the post-harvest management of the crops, and early disease and pest reduction and forewarning, and even weed control. So there are several aspects where the phenomics can be used. In the last almost about 1 decade, a lot of emphasis is being made on the phenomics, which is sensor-aided high throughput phenotyping, because the conventional phenotyping is labor, space, and time intensive. So it is with the speed at which we capture genotype information, we are unable to capture the phenotype information. So this creates a genotype-phenotype bottleneck in the analytical breeding. That is, once you establish a genotype-phenotype link, then you can easily use it in the breeding program. So besides that, we mostly do phenotyping at the endpoint, and so the final yield is interaction of let's say in rice, if you have 30 to 40,000 genes. So these genes interact with each other and interact with the environment, and ultimately we get the final phenotype. This interaction is constant, that is every worm in that is from genome, epigenome, transcriptome, proteome, and every worm interact with the environment, and it creates a lot of variation. So that's why the role of environment in phenotyping is very much important. And phenotyping at multiple time point is important. Multiple time point or the multiple different developmental phases of the plant is important to capture the genes that play important role in different phenological phases, so that we can enhance the genetic gain when you assemble all those things. So for these reasons, it is suggested that phenotyping is king and heritability is king by Roberto Tuberossi. So this phenomics almost it is evolved. The term was coined almost in several years back when the Human Genome Project was. What to do next? It is early 2000. And they, in that meeting, they coined the term phenomics, that in analogy to the genome, whether we can get the phenotype at the genome-wide scale. That is it possible to get that? So that's why the term the phenomics was coined, and the phenome refers to the phenotype as a whole set or full set of phenotype of an individual. That is expression of the genome as traits in a given environment. So the phenomics is the study of phenome in the genome-wide scale. So unlike the genomics, it is very difficult to by using phenomics to capture all the phenotype of a plant in a given environment, because of the limitation at present we have in measuring each and every trait at cellular level. The trait can vary from a change in metabolite to change in the just visible appearance of the plant, or change in the plant processes. Everything is a phenotype, but only we are present we are able to measure few phenotype which are like obviously visible, or some biochemical phenotype. That's what the technology at present we have. So this like, however it is, we can record more number of phenome data as compared to that of the conventional phenotype at a point of time, and we can observe the phenome change in different tissues and different part of the plant by using the high throughput approaches. So the high throughput phenomics, it uses non-invasive sensor and computational science to infer the data. The sensors that are used in the phenomics, they are of different kind. In general, we can put them into different categories. One is the group of sensors that measures the reflectance. So this any solar radiation or artificial light source, when falls on an object, we know that some of the light will be absorbed and some will be transmitted, some will be reflected. So the amount of light that is absorbed is depending upon the chemical composition and physical nature of the object. That is how cells are arranged, what is the composition, and what metabolites are there inside the cell. So depending upon this absorption is there. And then whatever is not absorbed, it will be transmitted, and some will be reflected. So the sensors that we have measures the visual sensor or the near infrared hyperspectral sensor, shortwave infrared hyperspectral sensor. These measures, they mainly are the reflectance from the leaf surface or the transmittance from the leaf surface. Whatever the light is reflected or transmitted, this light is captured by this camera, visual camera, or the VNAR camera, or shortwave infrared hyperspectral camera. So the other sensors, they use specific mechanism. One is the thermal cameras. They use the radiation emission. That is, each object above the absolute temperature, they emit a certain radiation in the range of 18 to 13 micrometer. If you capture that amount of radiation emitted, you can measure the tissue temperature. So thermal cameras use this principle to measure the tissue temperature, whereas the chlorophyll fluorescence camera, it uses a specific principle which I will explain later. And the laser based sensor, they mainly measure the volume or height. It is just based on the radiation, the laser that is emitted from a source that is reflected back. So depending upon where each beam of laser comes and hit the object and is reflected back, a 3D volume of the object is detected. Based on that, you can measure either the shape, volume, or the height, these kind of parameters. So these are broadly three kind of principle in general is like chlorophyll fluorescence, it uses a separate kind of principle. So this, although the earlier it was established like in the establishment of high throughput phenomics facilities, it started in around 2008, 9, 10, and earlier the facility initially it was established the controlled environment phenomics facilities, wherein the plants are grown in the greenhouse condition, and these plants are taken into the imaging unit where these cameras are there and the plants are imaged. This is kind of a whole body scan of plants. Like just when we do MRI, we just our like body part is put into the MRI, and then the reading is taken without any adverse effect on this. So similar way, these plants are subjected to the whole body scan, and the data is collected. So this kind of facility ICR has initiated initially in around 2010. So these facilities at IARI, CRIDA, NIASM, and IAHR Bangalore, these were established in different stages. Like in CRIDA, it was established in around 2014, and in the NIASM and IAHR, it was in 2015, and in ICR, it is in 2017. So these facilities in other institute, they have smaller facilities, and the IRA facility is one which is the largest with the multiple sensors and the multiple in terms of its capability as well as in terms of number of germplasms that you can see. The sensor, we are putting it into a fixed phenotyping room, and the plants move to the camera. The other approach is you can do it also in the natural field condition where the camera moves to the plant. One is the mobile platform, that is you have a fixed frame wherein the camera can move, or sometimes you keep a frame and the frame can move. One which was established in IRA as a fixed frame, that one that is there in the ES Bangalore and ICRISAT, where the frame can move along with the camera to different plants and take the image. So in this field kind of facility, you can also use load cell if you are growing plants on pots, so that you can also monitor the water use in the field condition. The recent one, which is relatively cheaper one, is the drone-based phenotyping, where the sensors, which I discussed with you earlier, all these sensors are placed in the drone, and then within a very short period of time, you can scan large area of field to get the phenotype data. So almost for last maybe 4-5 years, a lot of these activities are in progress in India. Many places people are trying to use this one, and with the promotion of government for drones in the precision agriculture, now a significant activities there in terms of precision agriculture. But for phenotyping, only limited efforts are being made by using this sensor. So since you are all in the virtual mode, I am unable to take you to our facility and show. I will show a brief video about the facility. So this facility was funded by National Agriculture Science Fund of ICAR, and it was dedicated to the nation in the name of Nanaji Deshmukh Plant Genomics Center, and by Honorable Prime Minister of India in 2017 October. So this video shows a brief about the center.
जलवायु परिवर्तन फसलों की उत्पादकता के लिए एक ज्वलंत समस्या है। भारतीय खाद्य सुरक्षा को बनाए रखने के लिए जलवायु अनुकूल फसल किस्में विकसित करना आवश्यक है। इस लक्ष्य की प्राप्ति के लिए भारतीय कृषि अनुसंधान संस्थान ने एक अत्याधुनिक एवं स्वचालित प्लांट फिनोमिक्स केंद्र की स्थापना की है। फिनोमिक्स एक आधुनिक बहुविषयक विज्ञान है, जिसमें लगभग वास्तविक समय में पौधों को बिना क्षति पहुंचाए सेंसर्स एवं कंप्यूटर की सहायता से लक्षण प्रारूपण किया जाता है। ये केंद्र हाईटेक नियंत्रित जलवायु वाले ग्रीन हाउस, गतिशील फील्ड कन्वेयर सिस्टम, स्वचालित भारोत्तोलन, उर्वरक एवं सिंचाई स्टेशन और विभिन्न इमेजिंग सेंसर से सुसज्जित है। यह केंद्र जलवायु अनुकूल फसल किस्में विकसित करने के लिए उपयोगी श्रेष्ठ जीन प्रारूपों एवं जीनों की पहचान करने के लिए प्रतिबद्ध है। पद्म विभूषण से सम्मानित स्वर्गीय श्री चंद्रिका दास अमृतराव देशमुख, जो एक महान सामाजिक कार्यकर्ता रहे हैं, उनके सम्मान में आज इस केंद्र को भारत के माननीय प्रधानमंत्री श्री नरेंद्र मोदी जी के द्वारा नानाजी देशमुख प्लांट फिनोमिक्स केंद्र के नाम से राष्ट्र को समर्पित किया जा रहा है।
I'm extremely happy to be here today to see this wonderful facility. I think when I was a young man student, we had so much trouble in recording accurate data, particularly in relation to phenotype, because phenotype is a product of the interaction between genotype and the environment. The environment has many, many parameters. We are in this facility, beautiful facility, and I'm happy to dedicate to Nanaji Deshmukh, whom I had known well, and who wanted the best in science to go to the forest for the villages. And that's the purpose also of IRI and ICR, to take the best in modern technology to small and marginal farmers. I hope this facility will be used not only to do a lot of theoretical work, but also in developing new kinds of phenotypes, which will help our farmers to produce more at less cost.
So this is about the facility, that short video. So we have sensors in the facility, a thermal sensor, which is IR sensor, which measures the thermal radiation emission from the plant in the range of 18 to 13 micrometer. And then we have a PS2 imaging sensor, that is chlorophyll fluorescence measurement system, imaging camera, and which takes the chlorophyll fluorescence to measure the quantum yield. And then we have the visual sensor, which is the most common camera, which is present in all the imaging facility. And then we have the NIR shoot image as well as NIR root image camera. And then we have the hyper spectral sensors. One is the near infrared range sensor, which takes observation from the 400 nanometer to 1000 nanometer wavelength. And then shortwave infrared hyperspectral camera, which measures the reflectance from the 1000 nanometer to 2500 nanometer. So these are number of image that is taken once the plant goes in. So total about 741 images are taken once the plant goes through these different imaging unit. So there is large amount of data is collected, and these are from the time of standardization to we have generated almost about 57 million images have been generated. And then we have large amount of watering data, that is how much water is used by the plant, that is whole plant transpiration is measured. And we have also genotyped all the germplasm or recombinant bloodline that we are using both. Mainly currently we are working in the rice and wheat. And for all this we have done both phenotyping and genotyping. And we have a dedicated team of scientists, of physiologists, where we have Dr. Dandapani, Dr. Sudhir, and then recently we have joined with Dr. Mahesh and Dr. Madhurima. So we have 4 different physiologists. And for rice, we collaborate with Dr. Gopal Krishnan and Ranjit. And wheat program is taken care of by Dr. Hari Krishna from the genetics division. And for many of the data analysis, the scientists from other disciplines are also involved. For especially hyperspectral data analysis and drone-based phenotyping, Dr. R.N. Sahu from agriculture physics, he is involved in the activity. And for several computer vision-based data analysis and use of the artificial intelligence machine learning, a team from ISRA, Dr. Alka Arora, Dr. Sudeep, and Dr. Sanjeev, Dr. Anil Rai, several teams are involved. So it is a multidisciplinary effort. Besides that, since it involves a lot of the image analysis and use of computer vision aided analysis, we also collaborate with IITs, IIT Delhi and IIT Mumbai. And we have multiple collaboration. They do all kind of analysis, because it is a multidisciplinary science, and the data analysis and interpretation, you need a team of multidisciplinary science. So this is just a simple work flow as we understand is given here. One is whatever, once you capture the image, these images, the raw image you get, and then the image is stored in our server. And then when we start image analysis, there is image pre-processing we do, where the image is enhanced. That is, use some techniques where the background versus foreground, there is a clear difference can be brought out. And in the next stage, image segmentation, where the background is removed, only foreground is kept. And then the next one is the feature extraction, use various algorithms to extract various features. These features may be the color, or it may be the shape, or it may be the number, or it may be the quantity of nitrogen present in it. Whatever feature is there, that feature is extracted depending upon the sensor that we use. And then the classification of the pixels, that is, if you want to know like leaf with X size, how many leaves are there, or what is the area of leaf that is thinnest, or what is the area of leaf that has X amount of water content, any kind of classification that is possible. So these are the different steps that are followed in the phenomics. And then we have the software that we have in our system, that is vendor provided software. It has a lot of complicated analysis, which you can select different algorithm to analyze each image, and then finally you get the output. And some of the now recently some softwares are available in online. So if you have any visual image, that also you can use it for analysis. So the visual image analysis is one of the most common image analysis that is used by all the high throughput phenomics people, and which can measure several traits which are related to agriculturally or economically important. That is, you can measure the biomass. You can measure the senescence rate. You can measure if you are doing stress phenotyping. You can measure the leaf rolling. You can capture the diurnal change in the leaf rolling. That is while during the daytime, how the leaf is rolling due to stress condition. And you can measure various other like caliper length and convex skull compactness. These are all affected during the stress, or it depends upon the plant architectural traits that can be measured by using the visual sensor. And this prediction of biomass is very critical, as the plant growth, which can be monitored by using this, either by using the growth rate, that is relative growth rate, or absolute growth rate, net assimilation rate. Everything can be calculated, which otherwise it is not possible doing it in the field condition for large set of genotype, because you need to take a destructive sample at multiple time point. And also it is useful to measure, accurately quantify any change. Either it is because of disease or because of stress, how the leaf area is changing, that can be monitored. So prediction of biomass, it is very accurate by using this approach. This one of my student Ilangovan has done this analysis, where he used a different 16 different machine learning algorithm to predict the biomass from the visual image data that we captured. So if we capture in case of rice as well as in wheat, another student has also done for the wheat. In both the cases, prediction of biomass till flowering stage, it is highly accurate. We can predict with an accuracy of almost about with R-square of more than 9 by using these machine learning models. However, once it goes enter into the flowering phase, after the grain development starts, the pixel volume will not change, only the weight changes. So in those conditions, the prediction accuracy decreases. Then we develop some other approaches to improve the biomass, which I will show in subsequent slide. So in the visual image analysis, not only the biomass, you can also measure the actual leaf area, which is again a very tedious process by conventional approach. Even you can measure the photosynthetic pigment to certain extent of accuracy, and it is like suitable for all the breeding application, because it can definitely rank the genotype very accurately, even if the exact chlorophyll content is not the same. So this can be done by using the simple visual image analysis. And as I told, once the panicle comes, assume that this is at the anthesis stage. So now this panicle has come after this no area is increasing. So in general, for image prediction, we use the projected pixels, how many pixels are there. Based on that, the biomass is calculated. So in this case, if we can count the number of years, and if we can count the number of seeds, then that information can be used to predict the yield and biomass. So towards this, Mr. Tanuj Mishra from ISRI Computer Science, he has done an analysis of using the wheat phenotype data that we captured. He has developed an algorithm, and this has been developed into a web-based application where you can count the number of VTR more accurately, and this accuracy is very high, almost 100% accuracy when you have images from different directions. That is, we take images in 3 different directions in our facility, so we can predict with more accuracy, almost about 99% accuracy. The number of years can be counted, and further what was also carried out, whether from the number of year and the area of the year, whether we can calculate the yield. So again, with this approach also we could achieve very high level of yield prediction accuracy. And again, the effort was made whether we can count the spikelets that are there in the year. So since we get very high resolution image in our facility, and it is possible even to count the number of spikelets that are present, and so these all these parameters will help enhance the accuracy of measuring the trait when you impose a different treatment, or when we have different genotype and we are doing screening. So this can be easily can be done. There are various other approaches also we tried. This is one of the startups which they are working. They used the YOLO approach to count the number of years and prediction of biomass. So various approaches are available which can be used to predict. And again, to calculate the senescence and by using the visible image analysis, we have developed a method so that different stages of stress or maturity, you can see how the senescence rate increases, and also you can see how the genotype differences are there in the rate of senescence. So this is under like drought stress condition. We can see HD2967 and versus the C306, how these changes are there. And this is another work which we have done in collaboration with IIT Mumbai. One of our ICR scientists who is Vishal, who is doing a PhD from there, he has done this work. So what he has done is, he has because we have the top image of the rice. He tried to measure the number of leaves. So it is useful in calculating the leaf area and the stress effect as if it is nitrogen stress or if it is water deficit stress. So it affects the number of leaf that emerge. So this he could predict with good amount of accuracy in the rice. So these are very useful in calculating the early vigor, and then we can see the genotype difference and how the genotype respond to the stress condition. So this can be possible by detection. And an effort was also made to see if we can identify some change in the leaf color, and that can be associated with the stress level, because the visible camera is one which is very cheaper, and if we can develop any method, that will be useful for various applications. So an attempt was made by Dr. Swati from IIT Delhi. So she has taken the visual data, and then she used lab color analysis to see if any of the bins in this color range, if it changes with reference to the relative water content. So these are the relative water content change in the plant, and then CES identified certain bins that changes as the stress level changes. So this, once we identify a specific bins that are associated with the stress induction, then it will be useful for the stress reduction. So these are all about the visual image analysis, which can also be used for disease detection and the detection of various mineral element availability. These are possible with the visual image analysis. So the next one is the NIR image camera that we have, which measures the reflectance from 900 to 1700 nanometer. In this wavelength, we have a major absorption peak of water, that is water absorbed with light around this range. So if we measure the NIR reflectance, we can calculate the leaf water status very easily. So by using this approach, we standardized method for measurement of relative water content and water status of the plant, which we conventionally do by using the method given by Bass and Weatherly, which takes 2 to 3 days to measure, and which is again very time consuming. And while collecting sample from the leaf and bringing it to the from the field to lab, it takes some time and measurement accuracies are less. So if we use an NIR camera, we can measure the water status of the plant with great accuracy. And mainly we use NIR camera for the measurement of water status. Then the chlorophyll fluorescence system is available. This camera can measure the chlorophyll fluorescence or the photosystem to yield. And I'm sure some of you might have used the fluoropen or the mini PAM to measure the chlorophyll fluorescence in the field condition. You do a dark adaptation and then you measure the quantum yield. And so for normal plant, it will be about 0.85, and if stress is there, it get reduced. So the similar approach is used, only this is here you get the data on each pixel basis. You get the entire plant, how many pixels are having high quantum yield, how much is having low quantum yield. So this is just 2 different plants are grown in the same pot and subjected to water deficit stress. Then after the end of the severe stress period, the image was taken. So we can visually see which plant is tolerant or sensitive, and that we can also quantify by using this approach. Hyperspectral imaging is one of the very important technique in the phenomics. So this can measure even the biochemical parameters by using this approach. So that's why this has given a lot of emphasis. So we have visual near infrared range camera, which measures the reflectance from 400 to 1000 nanometer, and then shortwave infrared camera, which measures the reflectance from 1000 to 2500 nanometer. So the beauty of this system is it measures the reflectance data in each wavelength is recorded. That is, if you want to know at around 500 nanometer what is the reflectance you can get, and 510 what is the reflectance you can get. So because each metabolite and the molecule, they have different absorption range, you can specifically get how the reflectance or the transmittance is changed in that particular point, and then you can get the easily measure the biomolecule. So this is just some examples are shown, like if you have this is Nagina 22 plant with different leaf relative water content. You can see how the spectra changes, and we normally use the spectral change at 1900 and 1450 for predicting the water status of the plant. This shows how the spectral change is there in the when the plant is subjected to nitrogen stress. Again, the different pigments, they have different the absorption range. So in this range, we can easily measure the reflectance to measure the various pigments that are present in the leaf, flowers, or in the fruits that is possible. So this is a study conducted by one of the students where we used the hyperspectral reflectance data to measure the water status of the plant. This is the stress index that he has calculated. So we can use these kind of indices very accurately to measure the stress differences in the different phenotype, or which can be associated with the relative water status. And we can also use this to measure the nitrogen status of the plant. And here the specific indices was developed to measure the nitrogen status. and we can see the distribution of the nitrogen level within the plant. The plants which are more greener leaves, it indicates high nitrogen level. The red color indicate the less nitrogen level. We can see the genotype differences in this, and we can also quantify this. And this one can also be accurately quantified. This is not from our study, but it is from the literature, where they have shown that it can use this kind of reflectance at different wavelength can be used to predict the nitrogen level. So these are different models that has been developed to predict the various nutrient level in the leaf of different soybean and maize plant from different studies. We have also attempted to measure the reducing sugar level, not reducing sugar level, and total sugar level in rice. So there is some progress, but this models needs to be further validated or further refined, as the current level of accuracy is only about 0.65 R square, or maybe between 0.65 to 0.7. By using this approach, we can also distinguish the genotype. So in this case, we have used the genotype can be distinguished based on the reflectance at different wavelengths. So these are different rice genotype which can be separated based on the reflectance. Similarly, this approach is also being used for disease detection. This is a study conducted by different researchers, and where you can distinguish the healthy leaf from the disease infected leaf. This is different diseases are shown here. You can distinguish. So a similar attempt was made by us. The study conducted by a master student in the agriculture physics, and in collaboration with plant pathology department. So this experiment was carried out to assess the rice blast disease by using the hyperspectral reflectance data. So here you can see various disease scores and how the reflectance changes there. And based on that, we narrow down to this particular wavelength range, where we can easily distinguish the this disease score and associate the disease score with the symptoms. So some model has been developed to associate the different indices have been developed to associate the disease level with the hyperspectral reflectance indices. So these models are being improved further for these are like the initial studies. So now again, a good big study is in progress to refine this. So I am sure that this will be very useful in detection of disease, not only for the phenotyping purpose, but also the precision agriculture purpose. The thermography, which is I think most commonly used by most of the breeders, you take the infrared gun and you measure the canopy temperature or canopy temperature depression. So in this case, the reflectance of the radiation emission is measured, so it is highly accurate method. Once you know the amount of radiation emitted, you can easily calculate the tissue temperature, and which is a very accurate method. Only by using this camera system, you will know what is the temperature of the panicle or what is the temperature of the tissue. So these are various cameras that we use, and maybe briefly I will also discuss with you about some examples of work that we are doing in our center. So one important aspect we wanted to address is the per drop, more drop. That is, under a field condition, it is very difficult to measure the water use efficiency unless otherwise you use lysimeter system, which is again highly laborious. You cannot do it with many genotypes. So hence we try to use the phenomic system for this purpose, because we have automated weighing and watering system. We can do weighing at multiple point of time. So it is possible to use this to measure the whole plant transpiration for over a period of time. So otherwise, by using IRGA, we measure only instantaneous transpiration rate, which are not very robust for phenotype. So in this case, when the plant takes up water, almost about 99% of the water is used in the transpiration, and only about 1% is used for the biomass accumulation. So, but this transpiration is very essential because this only facilitates the gas exchange and carbon dioxide uptake. So crop yield, it is defined as the total transpiration and water use efficiency multiplied by harvest index. This equation was given by Pasiero. But so far, the effort to use this in breeding is limited because it is the difficulty in measurement of these two traits, that is either the transpiration or the water use efficiency, which is not possible by using traditional approach. So people normally use these kind of pots. They grow the plant and then weigh the plant by using scale daily. So it is a highly laborious work. So that's why the progress is very limited. Since this facility, we can use this for easy measurement of the weighing and watering. We can do daily, and then it is easy. And then we grow the plant in the pots, and in the top 5 centimeter, we use polypropylene mulch. So there is no water that is lost directly from the soil. And all the water that is lost is moves through the plant only. By using this approach, we measured the like water use for this is one of the recombinant inbred line population derived from BVD 109 and IR 20, and similarly we have done for the genotypes that are the 3000 genotypes of rice. So we have used 2 different sets. For both we have done this kind of analysis, and we calculated various the image derived parameters. And then these are some of the trait that we used for QTL mapping. The large number of sets that we have used, and I just shown you some of the traits that are related to transpiration rate, some of the trait related to the transpiration efficiency and water use efficiency. Because in this, based on the image-based approach, without destroying, you daily measure what is the whole plant transpiration, and also you measure the biomass and the water, the leaf area, which is not possible in the like conventional approach of just only taking the weight, because we will not be able to account for the leaf area and other trait. So by using this trait, we calculated, we measured some of the data. Interesting data is there. So normally we assume that in the C3 plants, the stomata is closed in the night time, and the plant uses very little amount of water in the night. In contrast to that finding, we observed that the plant uses almost about 15 to 30% of the water in the night. Also, there is significant amount of genotype variation. Some genotypes they use about 15% water of the total transpiration. Some genotype they use about 30% water. So this is the kind of variation that we find in rice germplasm. So these are the 2 parents, and how the water use efficiency is difference is there for the whole plant, whole day water use efficiency, and for daytime water use efficiency and nighttime water use efficiency. And these are some of the transverse segregant lines that are identified, which has very high water use efficiency as compared to the drought tolerant BVD 109. So then we used kind of data, like large set of data we collected, and then the genotype information was used to map QTL. And since the data accuracy was very high, we could map some of the QTLs, which are highly significant QTL with very high phenotypic variation, as well as very high large score. And these are the number of QTL that we measured for various traits, and we measured one very important area on the chromosome 6 for various traits. And these are the different genes that we could identify by using because we know the marker interval, and we just saw what are the genes that are present in the map QTL interval. So we could see that many of the genes that are known in Arabidopsis, which are associated with the stomatal closure, stomatal development, and the abscisic acid pathway are localized in this one. So further validation is in progress. Like besides this mapping population, we have generated 6 other different mapping population based on the contrasting genotype that we identified by using the 3000 genome rice line phenotyping, and these are in F6 stage. So maybe in a year we will this coming season we will validate it, and I am sure this will give some validated QTL for enhancing water use efficiency. So similarly, the other important input that is important is the nitrogen. And if we take both wheat and rice, almost about 50% of the nitrogen used among the field crop is used by these 2 crops only. So enhancing nitrogen use efficiency is very important, because not only for enhancing the productivity, but also to minimize the environmental impact. So one of the interesting aspect again here also, we are always interested in transpiration also, that is water use efficiency also. So if you see this, this is the water use in the control condition, and this is the water use in the say in the nitrogen deficit condition. And the nitrogen deficit condition in general, both rice and wheat, they use more amount of water as compared to that of the nitrogen high nitrogen condition. So again, this is very important, like when we are doing the nitrogen management. And then we also actually measured the nitrogen content by using the manual method in addition to the phenotyping approach. And then we identified genotype that are having very high nitrogen uptake and utilization efficiency. These are the normally ROSI is one considered as one of the check for nitrogen uptake efficiency and nitrogen utilization efficiency. When compared to that, these are the mega varieties. They have a better nitrogen utilization uptake efficiency as well as utilization efficiency. And we have some of the genotypes that have really very high nitrogen uptake efficiency as well as nitrogen utilization efficiency as compared to that of the best varieties and best check that we have. So again, this is under different nitrogen deficit condition. Again, this kind of data was generated. And then we based on this, we have identified several genotypes from the 3000 genome line that are having both under control and stress condition, they have high nitrogen use efficiency ones. So now the selected ones we have also crossed to map to develop a real population. Again, the GWAS approach was used to identify some of the QTLs that are the market rate association that are associated with the nitrogen utilization efficiency and nitrogen uptake efficiency. So I think already I have taken about 55 minutes. Just in 5 minutes, I want to again show you some data on wheat. So this is
Okay, sir.
yeah, 5 minutes. So this one is on the wheat. Again, we used the germplasm line as well as the rails. So again, for both water use efficiency for yield as well as in biomass, we could identify lines which have very high water use efficiency. The C306 has a water use efficiency around this mean range only, but we could get many genotypes that are having very high water use efficiency both in the control condition as well as in the drought stress condition. So I think this is the same. This is the mapping population between the C306 and the HD 2967. And this one summary slide, just I want to show. So this is the amount of water that the genotypes use for grain production. So almost both the genotype, either C306 or 2967, they use almost similar amount of water for the grain production, and we have got some rills that are using less amount of water to produce each kg of grain. So under drought condition, this drought was imposed on the reproductive stage, that is after anthesis stage. So that's why the efficiency or the amount of water that used to produce per kg is more, because whatever water used in pre-anthesis stage, that was not exactly converted into grain. So that's why you see more amount of water under that. So it is depending upon the stage at which you. We also use
Kindly mute yourself.
with this you can use the sensors on the drone to take phenotype data. This is again a very efficient method for quickly phenotyping. And unfortunately you are not here, otherwise we have a very good lab on the drone developed by Dr. Salvo and IRI. So you could have seen.
Yes, sir. His lecture is also there, sir, on Monday.
Okay.
He will be delivering.
He will cover all this aspect about the drone-based phenotyping, which is again whatever genotype we have phenotyped in the phenomics, that is controlled environment condition. Again, they are grown in the field condition, and various stress indices are calculated to see how each this line is performing in the field condition. So I think this he will cover in his lecture in detail. So these are some of the data calculated, like actual genotype basis, and this is very useful in measuring either nitrogen deficiency or the water deficit stress based phenotyping. And besides that, you can also identify the rills that are similar to either parent or different from the parent. That also by using the spectral similarity, you can easily identify. That is just like by using genotype, you identify by using this approach. Also, you can identify by using the spectral similarity, you can identify the rills that are similar to either of the parent. That is possible. So to summarize, this precision phenomics is useful to reveal the genetic variability in protease efficiency. And from our study, we find a lot of variation in the total diurnal and nocturnal transpiration rate, both in the rice and wheat. And the nocturnal transpiration, we are now currently studying the basic mechanism, and also see if we can reduce some amount of nocturnal transpiration, so that which we think it is not totally useful, and it may be some amount of nocturnal transpiration may be essential, but not to the extent of the currently what we see. And drone-based phenotyping, I think it will be like more and more it is widely applicable, because the cost of establishment of that facility is relatively less. So now various other technologies are coming in, use of the machine learning and artificial intelligence. Various technologies are coming in, and only thing is you need the team of multidisciplinary scientists to do like from the phenotyping and take it to the breeding to develop a genotype, or to use this in the precision breeding. Just the last, what I want to say is, it is the question that gets you the Nobel Prize. After that, you just add lab coats, coffee, and air conditioning. So you need not have this very big facility in each and every place. Like, it's nothing that we don't have this facility. Only your research question and idea is important. Even with you can think of the alternative to each and everything, and we can do better phenotype. So I thank all of you for your passionate listening, almost for 1 hour. And just I thank the NISF funding agency and Director General ICR, Dr. Mahapatra, who played very key role in establishment of this facility, from the initiation of the project to establishment of the facility. And various committees that are contributed to the establishment of facility, and large number of students and scientists who did all the work that we have presented. Finally, I thank Dr. Jyoti and Dr. Kuldeep for giving me an opportunity, and also the director and PPGR, Dr. G.P. Singh, for giving me an opportunity to present this work before you. Thank you all.
Thank you very much, sir. Now, sir, if you permit, we can take a few questions if they have. So, dear participants, you can have some quick questions, or you can put in the chat box. So, sir has covered very quickly the topics on phenomics, because today in this era, the phenotyping is very much important. Now, genotyping is not that much difficult, but phenotyping, particularly for large number of germplasm and mapping population. So we need the use of high throughput phenotyping and phenomic tools. So anyone if wish to ask, so they can ask, or otherwise means they can put a mail to sir also, and he can reply later on.
Yeah, I think from this year we will be also opening this facility to other people. So maybe on charge basis. So
okay, sir. So all charge basis it will be on, sir, means per sample basis, per genotype basis.
Yeah, per genotype basis. Yeah, it will be open. So sometimes.
Okay. Okay, so this will okay. So regarding software, sir, I wanted that software there already means modeling has been done for predicting means phenotyping data from the image-based data. So here also, sir, you are developing your own model, sir?
Yeah,
because you are taking image data and also that manual data also being recorded for plant parts and all.
For most of the trait, we try to develop our own the model for validation.
Okay,
so,
sir.
Otherwise, some models are available in the the commercial software. Otherwise, like we develop for most of the things we validate in our facility also.
Okay, because in that commercial software that may not be validated in this population, no? Because here
for the specific crop it is not validated, and there will be a lot of variation. Better every time whatever we do, once we validate with the our own method, then that will be useful for our population as well as our kind of crop that we are using.
Okay, so that exercise is also going on, sir.
Yeah, that is for almost for all the trait we try to do. That is the first step we do before recording the large scale phenotype.
Okay, yeah, I saw for spikelets also.
Yeah,
so that is that can measure spikelets also.
Yeah, it can measure spikelet also, up to that level. This one PhD student worked on this to detect. One student worked on the spike detection and spike counting. Other PhD student he worked on the spikelet counting and how it can be used for the yield prediction.
Okay. So if there is no question, then I would like to thank sir, Dr. Viswanathan, sir. So sir, I would like to thank you on my behalf and on the behalf of organizing team and director and PGR, sir. So for accepting our invitation, because it was very high demanded topic by the participants. So we wanted this lecture. So in spite of, sir, your very busy schedule, sir, we agreed to deliver the lecture. So we thank you very much, sir, for covering all detailed account of phenomics facility, tools, and everything, sir, and how it is being measured like that. So thank you very much, sir, once again.
Thank you, Chodi. Thank you all the participants, and see you soon. Thank you, Chodi. See you.
Thank you, sir. Thank you, sir. Thank you very much, sir.
Thank you.
So dear participant, now I can see that our next speaker is also online. Dr. Rane, sir.
Yes, I am online. I was listening a bit to Dr. Viswanathan. Probably he must have made my job a bit easy. Shall I share my?
Sir, just a minute, sir. Just I will just repeat.
Yeah.
So dear participant, our next speaker is Jagdish Rane. So just I would like to just a minute, sir. I will just introduce. For that, I need to open the slide. Whether my screen is visible? Okay.
Yes, ma'am.
Okay. Okay.
It
Okay.
is not in PowerPoint mode, ma'am. It's okay.
Okay. Okay. Is it now?
Yes, ma'am. It's okay.
Okay. So, sir, we welcome you uh, for this training program, accepting our invitation. So, Dr. Jagdish Rane, he is now Director, ICR, Central Institute for Arid Horticulture. Sir was earlier at NI Baramati. So, sir, area of specialization is crop physiology, and he has great expertise in phenomics tools as well as high throughput phenotyping tools. And sir has worked for several techniques and research aspects like application of non-invasive tools and techniques to quantify plant response to soil moisture deficit, high temperature, water longing, and several other abiotic stresses. Then sir has designed phenotypic solution, affordable phenotyping solution to identify genes or QTS associated with abiotic stress tolerance. Then sir has also developed and applied phenomic tools for identification of appropriate bioregulators, which can mitigate stresses in plants. And sir has received various awards, fellowships, and recognition. For example, I'm mentioning few here: that Agriculture Innovation Award during 2020, Best Scientist Award of ICR NISM Institute during 2018-19. Sir has received Crawford Fund of Australia training during 2011. And he has been the scientific member of outstanding staff for the year 2009 at Columbia. Then he is fellow of Society for Advancement with Research also. And sir is member of several professional societies. Around 17 societies he is the member. And regarding publications, sir has published more than 70 research articles in the very international reputed journals with high impact factor. So, sir, I am stopping my slide, and sir, please present your presentation, sir. Once again, thank you.
Thank you, Jyoti, for that brief introduction and a generous introduction. Thanks a lot. And I would like to share the slides. You may have to tell whether I am audible.
Yes, sir.
And-
Very much audible, sir.
Okay, and then let me go to the first slide and make it visible for all of you. Is it visible?
No,
Visible,
sir.
sir?
Yes, sir.
Yes, sir.
Yes,
Perfect,
visible,
sir.
sir.
Okay,
sir.
thank you. Thank you. And just there is some obstruction at the top. I will hide that one.
It can move, sir. Meant with cursor, no. It can be
Yeah.
moved left and right.
Yeah, that's right. Okay. So good morning. Good afternoon to you all now. So the topic which was agreed upon by Jyoti and all the team who are organizing the training was affordable phenotyping for abiotic stress tolerance. Let me start with what I am going to talk about, a bit of content that I will tell you so that you can get prepared for that one for almost 40-45 minutes. Abiotic factors, that's what like you all know about. It's always better to start with what we all know about. Then phenotype and phenotyping, that also you may be knowing. The deficiency in conventional approach, one slide that I'm going to project. Modern approach to phenotype, several slides we have. Then I have some affordable phenomic slides for you, how like we can do the job, which high throughput expensive phenomics, what described by Dr. Vishwanathan a couple of minutes ago, can be done with some of the tools that you have in your hand that I am going to tell you. Then opportunity to translate knowledge into tools. That's the message that I'm going to give you when I talk about the way forward. Let me have the first slide. So we all know there are some abiotic factors. They are mentioned in even in our Vedas. So long back, 1,000 years ago, probably. So we all know that there are five factors: earth, water, fire, air, space. They all have been clubbed in Pancha Mahabhutas. All these factors, though they are not living, they influence each and every life on the earth. That includes even the plant. That's what, like our DG, when he was director of our institute at National Institute of Abiotic Stress Management, tried to put it in this type of slide, where he wanted to convey that air, water, soil, which all are non-living factors, along with the radiation that we can get, sun, everything influences the plant growth, and the plant is very much essential in terms of crop, in forestry, and the feed for animals, and also indirectly for the feeds for fisheries, which all make food for human being. So all these abiotic factors, they are very important, but unfortunate thing is when they are in excess or in deficit, they cause stress for plant and farmers. They live that abiotic stress are the age-old companions of farmers. So that is the concern. The more deep concern is that abiotic stresses are going to get aggravated due to the climate change. I need not explain, because this picture itself explains 100 stories about it. Either cyclone, heavy rains, heat wave, flood, drought, all of them are like sources of abiotic stresses. They are going to increase in the frequency and also the intensity. That has been highlighted in IPCC report 2021, and also several articles written by Director General ICR, Dr. Patak. So when we talk about genetic resources for harsh agroecologies, including rain-fed agroecologies, we may have to reimagine a quest for plant features. So those features can be grouped into 3 groups, maybe more if you imagine. First one is adaptation to extremes: soil moisture deficit, high temperature, nutrient deficit, salinity, waterlogging. You may add few more things, including the high metals toxicity and so on. Then second group of features, high input use efficiency for applied water, available radiation, energy used in agriculture, labor, stress alleviators that are being talked a lot nowadays. The third feature is eco-friendliness, crop management compatible, high carbon sequestration, zero emissions, rhizospheric microbe compatibility, all those things you can add there. So if you want to have all these 3 group of features to make the plant climate smart, then we talk about plant performance under both optimum and non-optimum condition, under natural as well as managed condition. They should perform better, and that's possible if they have inherent capacity to do it. Inherent capacity to do that one is governed by the gene. That's the reason we talk about genetic resources, which are all available, and features which can be brought into agronomically important crops or varieties which are prevailing in farmers' field. So when we talk about bringing the traits, studying the genotype and also phenotype, we talk about genotype into environment interaction. But nowadays, the management component, that which creates a microclimate, that's also very, very critical. So when we talk about the management component, then that adds additional feature, additional agroecologies that we have to talk about, and additional design or features are essential to make climate smart crops, that we have to keep it in mind. Can we do it with conventional approach? If you look into the strength, weakness, opportunities, and threats of the conventional approach, the beautiful part of it is the strength, which focuses on yield potential. The success that we got by this approach is tremendous, and that's resulted in tremendous success of the Green Revolution. Focus on yield, focus on disease resistance, focus on quality, that have yielded lot of dividends. But the weakness is they are not robust enough to address abiotic stress rates, resource use efficiency, or management compatibility. They are not designed for that. Then climate smartness, which we are talking a lot nowadays. Then opportunities, we can integrate this conventional approach, which is inevitable, with all those modern approaches, that what we call as omics approaches. compatibility with emerging NRM solutions for climate smart agriculture. We have to come out with the ideas. I hope I am audible, no? If
Yes,
hello.
sir. Yes, sir.
Yeah,
Audible,
okay.
sir.
Okay,
Audible.
thank you.
Mhm.
So the threat is when we talk about abiotic stress, if it is a beyond control, beyond limit, many of these things may not work for that particular season, but definitely it will be useful for the next favorable season. With that optimism, we have to carry out all these research activities. We have to answer many of the research questions, keeping in mind adaptation to extremity, resource use efficiency, and eco-friendliness to reach the next height in achieving food sufficiency and food security for the nation. So, if so, what should be the reimagined route for genetic resources? So, we are talking about agroclimatic regions in India. They are very broad. So, such a big country, if you look into those fractions which have been projected there, it is something like 15 agroclimatic regions, or they have been further divided into few more. But if you look into the type of soil and type of climate or weather in each of the location we experience, they can give hundreds and thousands of combination. If that is the case, we have to have a designed crop for each of these combinations. So this approach should be run in parallel with the current trending from seed trade to the gene trade in the context of adaptation to extreme, resource use efficiency, and eco-friendliness to achieve the target. And here again, genes will be the key for crop design. So when we talk about the abiotic stresses, it is not a single abiotic stress, but it is the combination. That's another challenge that we have to look into. All those abiotic stresses originating from atmospheric or due to water or the soil have been listed on this slide. That is just to say that it is not a simple task, but we have to look into that. When we talk about gene function, so we are relating the gene to the features of the plant organs and all. That is very much evident from Mendel's experiments and linking traits with the gene and all, and phenotype and the genotype. In addition to the environment we talk about, lot of literature is emerging and lot of books are available now that have been projected here. One is phenotyping for plant breeding, then high throughput phenotyping in plants, and many more. Then phenotype and phenotyping, I need not probably again repeat it because Vishwanathan must have explained, but it is better to tell this one in the context of affordable phenomics. So this is what the slide of just copied from one of the article explaining Mendel's work. We all know we have to characterize the plant responses in terms of morphology, physiology, biochemistry, and many more things. And we call it as a phenotype, routinely carried out to differentiate genotypes on the basis of responses to imposed treatments. That's what the phenotyping we talk about. Why phenotyping for crop improvement? Every one of us know to improve productivity of crop, quality of crop, tolerance to biotic and abiotic stresses, that is based on the associated plant traits. And this all is possible when traits are governed by inheritable genes. So all focus was there on the gene. People felt that once the genes are sequenced, we will be able to decipher many of the mechanisms, many of the traits and all. And that ambition and that like dream started with sequencing the human genome long back in somewhere in 2001, but it was a huge task for 500 people across the world, and more than $500 million were spent on that one, and it took almost 5 years or more for that. But to do the same job with it is possible to do that job within an hour or within a day with the tools available now to sequence the gene with next generation sequencing and all. So many things are available. What it means, the understanding the gene is like now become very easy, but its function, if we come to know, we can achieve the target of designing the crop. If you take Arabidopsis as an example, lot of gene information is available, but even 30% of that gene function is not known. So this is case with the simple plant like Arabidopsis. But if you talk about crop which we are growing for food, that is still complex. There's a lot of challenge. And hence, to elucidate the function, we are looking into the forward genetics and reverse genetics. Forward genetics, which starts with the known phenotype and ends in discovery of the gene, and the reverse genetics starts with the known gene, which are like derived from crops like Arabidopsis, and ending with the phenotype. So both of them that need phenotypic information. The critical questions to be answered here: can we get benefit of the gene with known functions transferred to crop plants? What are the functions of the rest of the genes? If you alter them and look into the phenotype, are they useful to improve crop plants? All those questions that we have to answer. That's where the evolution of phenomics as a new complementary science to other omics like genomics emerged. People started talking about screening large number of genotypes where the genes are altered by mutation. They were in thousands and 10,000, and by manually, it was really impossible for people who thought about it, and they thought machine should be engaged, machine vision to be engaged, and data analysis, data laboratory information system had to be developed. That's where the phenomics evolved early in 2000. With the rapid development of genotyping technology, the general term of phenomics was widely adopted to cover automated plant screening work. So this is one of the example of treadmill or the high throughput phenomics, which was developed earlier in Belgium and now taken over by the BASF. So the reason given for those heavy investment, millions of dollars, in Indian terms, 10 and 25 crores of rupees for that type of establishment, was defended by telling that conventional approaches are not so successful in giving additional genetic gain, either in wheat, rice, or many of the crops. That was one of the reason. The second one was abiotic stress, which are going to get amplified because of climate change. So importance of phenomics and the vision about this particular science was like reflected in one of the review in Nature review very nicely 10 years ago. Since then, lot of progress has been made. So if you look into the plant phenomics and non-invasive feature of it, it is a niche for application of photonics. What the photonics is? Photonics tells about the plant's capacity to reflect or absorb or pass through some of the radiations. When we talk about radiation, it is electromagnetic spectrum, the part of the electromagnetic spectrum. When we talk about part of the electromagnetic spectrum, we are very well acquainted with the visible part of electromagnetic spectrum, that is 300 to 700 nanometer around, which we all can see because the sensors in our retina, they can sense one. But there are many radiation which cannot be seen by human eyes, but every object, it reflects or it behaves in a different way with respect to transmission of this radiation or absorbance, and that is the typical signature of that particular object. So that could be sensed with sensors, and those sensors are embedded in imaging system, and with that imaging system, lot of traits, particularly in case of plants, can be quantified. So for example, color images, like what you call visible range of electromagnetic spectrum, if we engage it in imaging system, we can look into plant area, mass, then leaf senescence, so on, which even Viswanathan also must have talked about. Then near infrared imaging, tissue water content, he must have explained. Then infrared imaging, fluorescence imaging. In addition to that one, X-ray images, high resolution NMR, terahertz imaging, people are talking about. So why all these features? Why all these imaging system? We want to know about plant features, about at cellular, tissue level, organ level, whole plant level, and canopy level. But when we talk about high throughput, screening large number of genotypes, then we are designing tools to capture the external features to understand the internal phenotypes. So external phenotypes and internal phenotypes and large number of genotypes, all these are the key words that we are talking in phenomics. And Zhao et al. 2019 the paper published in Conscious in Plant Science. You can go through it and you will get a lot of information about these concepts. When we talk about external phenotype, then we are talking about plant shoot traits and also root traits to understand what exactly happening inside the plant indirectly. That becomes that job that makes the job easy when we talk about large number of genotypes. Then yield defining traits, when we talk about shoot traits and plasticity, influence architecture, fertility, shoot biomass, photosynthesis, stomatal movements, that is water relations, assimilate loading and partitioning, particularly in the context of abiotic stresses, senescence timing, how fast the senescence take place. All these are the features that some of the features that we have to look into when we talk about abiotic stress response of the plant. Can we employ phenomics tools to do that one? That is really very, very important. Here we talk about high throughput, but bit expensive, not bit expensive a lot, a type of instruments, and they emerged in European countries, and also they adopted by Australians and more or less all the labs throughout the world. And these are one of the example which I could see some 10 years ago. That is a greenhouse where every pot was moving, and robotics which can see the plants from all the angles and capture the image. That's one side in the laboratory condition and even in field condition. Instruments to look into the movement of even metabolites within the shoot, they were designed in Hulich, a very advanced lab in Germany. So how it started, phenomics, that is concept was there in 1911, Belgium crop design, Australian plant phenomics, German plant phenomics, European plant phenotype network, IPPN, International Plant Phenomics Network. All these are the keywords. If you put inside the Google, you will get a lot of information. Now in 2019 to 2022, if you look into multi-scale phenomics, huge amount of data generated by the machines, and the need of big data analysis, artificial intelligence, machine learning algorithms to look into, they are all emerging, and now it has become truly a interdisciplinary science. So we do have a high throughput phenotyping facility at 4 places that must have been explained in the previous talk at ICRISAT and US Bangalore. That much of that is focused on the controlled environmental, but we are talking about field phenotyping platform also. Broadly, they are using thermal, multispectral, and hyperspectral image sensors. And if you look into the shoot system architecture, we are talking about the visible light imaging to capture particularly morphological traits, digital biomass, height assessment, and fluorescence imaging, particularly to understand photosynthetic status of plant under abiotic stress condition or given environment. And there are high throughput phenomics which features sensors to plant movement that's under controlled condition. I told about visible light, chlorophyll, fluorescence, infrared camera, and all. The phenotyping for plant geometry, growth, and biomass, counting growth stages, vegetation indices, and all that was involved. Then there is another one, the plant screen, that is also high throughput, particularly for crops like Arabidopsis, which are being used in Cairo, Canberra a lot for looking into the growth rate, nutrient management, photosynthesis, pathogen interaction even, and trait identification, and also chemical screening. So high throughput plant phenomics, controlled environment. This is one I was trying to project, trait mill, which was developed in crop design, that you can look into this website, www.wps.eu. So if you look into all those fractions or sections of electromagnetic spectrum, the machine vision have been designed here for each of them. So this is one of the example where each and every wavelength of the electromagnetic spectrum can be explored to understand plant responses to abiotic stresses. Companies are available for that one. Tools are also available, but they are still not so much affordable. In years to come, they become affordable. And when it comes to root system architecture, we have fantastic instrument now. They are engaging X-ray CT technique to look into the root. which gives information about the root growth, 3-dimensional, 2-dimensional, whatever you are talking about, without disturbing the soil. And medium throughput facility have been developed in countries like England and also Ireland. In this system, you can just see how the potato grows without removing the roots, extracting the root from the soil. It is really very, very clear. And that type of sophisticated instrumentation is available for phenotyping, but they are costly. In addition to the X-ray, CT scan, MRI, magnetic resonance imaging, that's also really very costly. Positron emission detectors, they are being engaged in understanding the root system. So they all are capable of giving us additional information, additional insights into the rhizospheric mechanisms without disturbing the soil. We started exploring the opportunities at NIAS with the phenomics establishment there, with target delineation, adaptation, and mitigation, all that in our mind. So particularly when we talked about adaptation, physiological manifestation, biggest challenge for us to get into that system and make best use of it, to understand how it works and what all its components have to be optimized. The components like steps to be optimized there to use all these phenomics facility was imaging unit configuration, image analysis configuration, and data acquisition and inference. When we talk about image unit configuration, how to capture the image in such a way that there should not be a background noise. And then image analysis configuration that is customized, that is given by the LamnaTech, that is specific to the LamnaTech only, and converting the image into the data set, then the mining of the data. Once the data in huge quantity in Excel, it runs into 10,000 or more than rows, like sometimes 60-80,000 rows, like we could handle with 500 columns. So many data it gives, and converting that data into meaningful illustrations and all, that was a challenge. It took almost 2 to 3 years. Since 2016 we could do that one, and ultimately we could publish high quality journals with indicating some of the novel traits like water use efficiency traits per unit of what you call relative growth rate per unit of water use indices we derived, and we could identify the genotypes there, promising genotypes. We went with almost every day's observations with all those visible camera, around a dozen visible shape features could be derived from that, from 3 angles, that is 42 parameters were there on one side, and actual data set of biomass was on another side. We just tried to engage some of the machine learning algorithms, 8 machine learning algorithms, to predict the biomass based on these all more than a dozen parameters from 3 angles, and we got a very good correlation between the predicted and actual value that we use for deriving the water use index with growth rate calculated for every day based on those models. Then plant phenomics, affordable. So all these phenomics facility which runs into crows, that they are not really affordable, every one of us know. And even if you all want to use them, and if you come at a time, then also it is not possible. Then why not to look into the affordable phenomics? That was dream even before this establishment. Phenomics was installed at different locations. We came out with our own design of a greenhouse at Indian Institute of Heat and Bali, where the roof as well as the side walls automatically they open when we don't want high temperature, but only at the time of grain development. If you want the high temperature, we can close them. And then planting method and data collection with whatever the conventional data that we take, that all were optimized, and we could publish a nice paper in crop sciences journal there that will be available for you. And one of the affordable phenomics designed by late Professor Uday Kumar needs a lot of appreciation. It gives an idea about how to screen water use efficiency in crops like rice, and lot of information has been generated by Dr. Uday Kumar's team, led by Sheshashai at the US Bangalore. You can go through that one. Then exploring non-invasive features of it. When we talk about non-invasive feature, then RGB color is one of the tool. Red, green, blue pixels of each of those images that will give lot of information about how the plant senesce, how the leaf lose their chlorophyll content. That is possible to do that one because all these pigments, including the chlorophyll, they have their typical absorption spectra. So that was the concept when we talked about implementing this one in screening soybean soybean bradyrhizobium strain for their efficacy to support growth, development, yield under water stress condition. And we could publish that paper. Initially, we came out with all those models, the validation that can we quantify the green color, can we quantify the blue color and red color. And based on that, we designed some of the software, and we analyze all those images, visible camera, that is normal digital camera we engage, and also IR camera images also we could analyze, and we could prove that this type of approach is possible. It is possible to use those approaches in screening the genotypic response and identifying the promising strains. That was the first attempt that we could do. And we had a separate chlorophyll fluorescence imaging system that cost around 10 to 12 lakh. I cannot say that everyone can afford that one, but it is not as costly as phenomics tools. Even if you have that one, that's one of the fantastic, wonderful tool to understand plant responses to different abiotic stresses. You all know that only part of the radiation, solar radiation, absorbed by the leaves, they are utilized for photosynthesis. The rest of the radiation get emitted either in the form of heat or in the form of chlorophyll fluorescence. The scientists have designed to capture that chlorophyll fluorescence with the assumption that healthy plant will not emit so much chlorophyll fluorescence, but non-healthy plants affected by stresses, they can emit more chlorophyll fluorescence. But the story is not so simple. Lot of formulas, lot of theories are involved in that one, and based on that, it is possible to quantify how plants respond, and what is the differences between or genetic variation among the genotypes. How to get that one with the parameters like quantum efficiency and all that have been all optimized. We try to optimize some of the methods with out of box thoughts, not only the leaves but also ear of feet that we wanted to try, with the hypothesis that durum is more tolerant as compared to the oestium wheat because it might be having better chlorophyll fluorescence efficiency even under desiccating conditions. Desiccation is the result of drought. That was the hypothesis. We tried to apply that one, and we screened the genotypes, some of the genotypes, and we could prove that truly the chlorophyll fluorescence of durum was found to be more efficient as compared to the oestium one, and that we could publish. And based on the demand, then we tried to apply this one even for the fruit crops, and we could show that some of the fruit crops, including the dragon fruit, they have more tolerant photosystem to efficiency as compared to other fruit crops, and that could be published again in Scientia Horticulture. So we talk about the surrogate traits when we talk about plant phenomics. Instead of plant height, plant leaf length, and so on, we take the features of those segmented what you call images, and there are a lot many that we wanted to try, but all the time, again, we just it's I will tell this one later. Okay, we try to include those shape features as a surrogate traits. I will tell you in the next slide. This interrupting slide came in between. But whenever we talk about phenotyping, then we should keep the mechanisms in mind. For example, drought coping up, drought tolerance, when we talk about escape, dehydration avoidance, dehydration tolerance and all. When we talk about drought escape, then we talk about seasonal escape. So we talk about early varieties which can escape the terminal drought or terminal high temperature and all. But there is a phenomena of diurnal escape also. When there is, if you take the 12 hours the daytime or whole 24 hours, plant behave in a different way. Plant has a capacity to close its stomata when there is a lot of demand for transpiration, the loss of water, keeping its canopy air cooler or something like that. They behave in a different way. If a plant is capable of transpiring, keeping its stomata open for what you call relatively low temperature hours of the day, either in the morning or in the afternoon, like in the evening. So that plant probably can perform better because it has to close its stomata to save water in the afternoon, immediately around 10 or 12 p.m. So if that is the case, can we like capture that trait in large number of genotypes? So we could prove that yes, it is possible to do it with the infrared imaging system, which tells about the canopy temperature or leaf temperature, and we can program that one to capture the leaf temperature every minute or every half a minute. And based on those huge set of data, we can say which one is more efficient. That we could prove. And that's what we wanted to do either even at CH with the rice transgenic lines and also soybean in field condition. Then identifying dehydration tolerant lines out of thousands of lines, that's what we employed there. And no, this is dehydration tolerance I'm talking about. The previous one was escape I was talking about. Then dehydration avoidance also can be talked about by infrared imaging system. How plant keep itself wet even when there is no sufficient moisture under in soil. That is the dehydration avoidance. That is extracting more and more water from the deeper layer. But when we talk about dehydration tolerance and osmotic adjustment and all, that is one of the factor. What I want to say here is, if I challenge any one of you that you differentiate the response of the plant to stress, then you go with all those gadgets of plant physiology, including photosynthesis meter, in relative water content, and so on, and you will tell that, oh, this has got better photosynthesis, better relative water content, cooler canopy, and so on. You are going to tell. But if the water content, like tissue water content, is sufficient in the leaves, then automatically all the physiological parameter will be good. So you need not go for all those parameters. What we really need is if there is 50% reduction in tissue water content of any of the plant, either it is annual or it is horticultural crops, and if any one of the candidate is maintaining better photosynthesis as compared to others, then we can say that that has got dehydration tolerance, true tolerance to abiotic stress. So what I want to say is, I want to measure all these parameters, physiological parameters, in a condition when it is 50%, like tissue water content is 50%. It is not that relative water content is so much, and so on. It is actually, I want that particular dehydration level common to all the genotypes. Is it possible? That is the big question. So if you look into the possibility of engaging NIR, near infrared radiation, it is capable of revealing the soil moisture content of leaves or plant at any given point of time non-invasively. Based on that, we can model what could be the moisture level at any given point of time, and what was the chlorophyll fluorescence imaging data that could reveal about photosynthetic efficiency. Okay, so some of the slides got mixed up. So with NIR, like we talked about excised leaf water loss as one of the trait. And that also depends on the context in which you are talking about. But it is possible to look into this one with NIR imaging non-invasively. That could be proved. And there was quite a good genetic variation here. Now I will take up features of climate smart crops. Okay, how to design. Let us start with the questions. Remember the last question, last slide projected by the Vishwanathan. That's the question which makes difference. Once you like wear all that lab coat and all those things, let us start with some questions now. So smart crop rapidly cover ground to reduce evaporative loss. Okay, so loss of water, soil moisture through direct evaporation, if you prevent, and if make the plan to prevent that one, then there'll be at least partly will be successful in utilizing rest of the soil moisture for plant growth and all. How to identify them? How to identify them is the question. That is how plant cover the ground rapidly. Is it possible to do that one? Yes. If you take the picture from the top view regularly, how fast they can cover the ground, that itself will convey the message. We did some of the exercise, tried to develop some of the tools with simple imaging system. Okay, so that is possible. Here exactly I am talking about affordable. It is affordable. Then smart crop sense slowly even under stress condition. How to identify? Already I told you that it is possible to judge which is yellow, which is green by engaging red, green, blue pixels. That's what we did. And we generated some of the data. We tried to like derive some of the indices and try to relate it with the actual chlorophyll content measure. And we got quite a good correlation. That is R square is equal to 0.87. This can be done by any one of you, but some precaution has to be taken. Smart crop rapidly recover from stress. How to identify that one? What I want to see? I want to see how they rapidly recover. So if you look into this particular slide, probably I must have projected it in my several previous lectures. So it is possible to look into the genotype which recover faster as compared to others when they are allowed to wilt total. It is possible to do that one. I'm projecting here only the two genotypes here, but it is possible to look into the shape features of many of the genotypes, and that is possible in plant phenomics, but we have made it possible even through other affordable tools that I am going to tell you. Then when we talk about this possible, and when we talk about like that, all those circles or whatever the shape that I have given, that is called as convex hull, one of the shape features, but we have 12 features or more than that can be derived, particularly with the tool of plant phenomics. But if you engage the JImage tool that is open source, there also will get lot of parameter. Okay, so based on that one, you can just talk about how they are related and how they can explain the tolerance to drought or any other abiotic stresses that is possible. So that was the picture which we took, and I tried to project the shape based on the high throughput phenomics. One of the MSc student, he came out with his own tool by using smartphone and capturing the images from top as well as from the side views, and he tried to compare some of the derived parameter from high throughput, that is the HTP, and affordable phenotyping tool APT, and we could obtain 0.97, 0.92, 0.88 correlation. So that means it is possible to engage some of the tools that you have very much in your hand, that is a smart mobile. Then out of way traits, so you should think of, like we thought that the pedicel which links the pod and the stem, that the main shoot, has to play some role in pod filling under abiotic stress, that is the depleting soil moisture condition. If to prove that hypothesis, I have to look into the pod features, how to like phenotype that the not pod, the pedicel feature. That was done by another student, MSc student. That's a very short experiment, and he screened large number, like around 25 genotypes, and for each of the genotypes 10 and 25 like pods and pedicels, and he could prove that the pedicel periphery that has positive correlation with the grain size, that indirectly indicates that the pedicel has to play some role. Can we phenotype for this particular trait now and identify the gene? That is the next step. And this is virtually, it is an affordable phenotyping, believe me. And he tried to like compare lot many features derived from the image analysis by image J. Any one of you at the end, like if you are interested, one day like I will explain that how it can be done, that is possible to do it. Okay, I am just giving a overview and what all possible things. Then this is prediction of seed yield by using pod images. This we tried in Greengram, simple mobile camera, and then we could extract lot many pod features, and this was the workflow, that acquisition of the pod images, a manual counting of the seeds on one side, weighing of the grains per pod, acquisition of seed images for analysis, image analysis for both pod and seed, then relating digital data with observed data, that we could do it. And we could like correlate a different manually measured parameter with the digital parameter. It came up very well, and that one of the paper is coming up, that boy is writing it, and that was quite a good correlation between pod length with the seeds per pod and also seeds weight per pod. This is all like how you analyze the image, how you capture the image, how you conceive the traits, novel traits that you are talking about. And phenotyping grains and spikes in cereals, that also has been like now gaining more importance. X-ray techniques and lot many other techniques are being used to look into the each and every grain of a spike. But what we did is simple, like we extracted the grain of each spike, and then we started to capture all of them. And again, like we went for image J, and each and every grain of the spike of so many genotypes, like we tried, and we got that image, the size. And then we derived a factor called deviation from normal when we talk about the distribution of size, to say that is an indirect measurement of intra-spike assimilation variation, deviation, what you call within the spike, how much deviation is there from the top grain, bottom grain, and like a middle grain. Okay. So that's what we wanted to calculate. So that we worked out, and we derived some of the parameters like width, length, perimeter, area, and the deviation within the spike. Hello? Yeah. And
No, sir, carry on.
yeah, we went for the standard deviation of grain width, length, area, perimeter to differentiate the genotypes. Okay. And yes, Jyoti, can you like mute them? If there are some questions, I can answer. But if there are no questions at the end, like we can answer. Yeah. So we look into the
ऑफिस से वो मतलब गुड़देव के पास में एक लकी रेस्टोरेंट है, फिर आप इधर आ जाए.
Yes, thank you for muting. So we like first validated whether it is possible to get the actual length or width of the grain, each of the grain, with all those image parameters we are deriving, and then onwards we could do it. And we did lot of analysis there, and part of the story was published in one of the article in agronomy journal there. Then smart crop established their route rapidly. Can we identify? Our plant phenomics facility was not designed for that. One of the student came and he started designing some of the things, and the way like putting all those in test tubes and transparent media, and we could come out with a protocol to look into the growth of the root in test tube itself. It can be done for the seedling stage, and we can differentiate the genotypes. That is again a kind of example of affordable phenotyping tool, rather than going for another like phenomics establishment with crores of rupees. Then smart crop retain pollen activities even under stress. Can we identify them? That's the question. And it is possible, even if it is possible with some affordable tools like a fold scope, we could look into the pollen viability here, and we could publish that one in South African Journal of Botany. You can go through it, and we can also look into pollen tube growth with that particular instrument. You can just look into possibility of doing that one. And if you have any like questions about that one, I will try to answer. Nowadays that fold scope are available even in market, and they are not so costly. Then smart crop
Which
have less.
instrument, sir? Which instrument it was?
Hello. This is,
Holoscope.
it is called fold scope.
Hold scope. Okay.
Fold,
Hold
fold.
scope. Hold
Fold
scope.
scope.
Okay.
Yeah.
Okay.
Oh, fold scope. That's what, like, it's on the right side. You can see it is a paper microscope. You can put it in your pocket or purse also.
Okay. Okay.
So with that, we could do that one. Okay. Then smart crop have less footprints. That is footprints of abiotic stress on seeds. That's one example I told you that should have gone there. And this is about phenotyping waterlogging tolerance. When we talk about waterlogging or submergence, then there are a lot many things happen inside the plant. So can we look into that one? One of the things is about the root, adventitious root or roots in response to waterlogging. Can we phenotype them? One of the student is like picking up that got mixed up again. Oh, I missed that slide. Okay, I could have shown that one. Okay, that we designed some of the tools to look into waterlogging induced roots, and that's again MSc student. He's still working on that one. Okay, if I get the time, I will show you how we could do that one. And low cost field phenotyping protocol. When it comes to, I'm really proud to say that institute like NISM could like come out with its own tool to look into the plant responses even under field condition. The idea is to make this tool automatically move in the field, capture the image, start with only the digital camera, simple digital camera, or even your mobile camera also that can be engaged here. And the principle is bit simple here. It will run on the track and it reads the barcode of that particular plot, and that sends the signals to the camera. Camera captures the picture in the name of that barcode. Barcode has the name of that genotype and name of the plot and name of the treatment. And you can capture so many pictures. Once you capture that picture, you have to segment it. You have to remove all the background. Then only you can use that image for further analysis. This is called as image analysis. That also we could do with imaging on our own tool. And then we analyzed, we derived lot many parameters related to that, including shape feature and also the RGB features to look into the leaf senescence. And this can be used and this can be made available for many of you who are in breeding or phenotyping, genotyping, QTL identification, gene identification. That's what the dream is. How it works, that I will give you a picture. I am not sure this video is visible or not. Jyoti, is it visible?
Yes, sir. Visible, sir. Visible, sir.
Yeah, this machine moves. This machine moves in the field. Once it reads the barcode, it stops and it allows the camera to capture the image. Image is captured in the name of that particular genotype, particular replication, and all that, and it saves there. And it keeps on moving. And if needed, you can run this one even under night condition if you want to look into the features. But you have to decide.
Just.
Okay, this is okay. So there are some disturbances.
Yes, sir. I removed, sir. I
Okay,
removed.
okay. Let us go to the next one. Then all that we are going to put in the name of imaging scientific hints about resilience to abiotic stress. That is the Ishara's model one that we put. That includes image capture, image analysis, database, and inference and all. And that will be able to give some big package for the breeders, and that will be a nice example of affordable phenotyping for Indian plant breeders, either in ICR institutes or even in SAUs and other institutes. So way forward, when we talk about, we should remember that future profit. 1 minute. Future profit from crop improvement to get leverage from advances in sciences. That we talk about quality, stress resistance to biotic and abiotic factors, then yield potential. That is not just sufficient. That has to be like divided, whatever it may be, you put a formula there. That's the function. Again, a cost to farmers, cost to environment, then the sustainability. And that's what like we are preparing for the future climates. That's what we have to remember. And science can play a very important role. Which genotype is better than others? That's the question. genes relevant to stress tolerance. That is really very important, hence the phenotyping there. We have to look into the mechanisms of tolerance, morphological adaptation to stresses, promising genetic stocks, new genetic variations, either created by mutation or nowadays we are talking about gene editing and all. All those are the plant types or what you call genetic resources. They have to be phenotyped for relevant traits and relevant genes. And we must remember this factor. That's a lot of variation in agroecologies that we are talking about. It should go with the agroecologies rather than agroclimatic regions. That will give you lot many combination of agroecologies and hence lot many possibilities of crop design, and each crop design has to be having a set of traits, and we have to design plant phenomic solution accordingly. Then this is really disturbing a lot. Inflorescence architecture, it is possible to look into affordable phenotyping tools. And shoot biomass, it is possible to capture even with your mobile or smartphone. Photosynthesis and all that needs chlorophyll fluorescence imaging system. They are in a few lakhs. The instrumental movements and density regulation that can be captured indirectly by IR camera, assimilate loading and partitioning and all. I didn't talk about hyperspectral imaging that is possible to this particular parameter can be measured with that one. That is a bit costly. Then senescence timing, they all are important. Okay, so then other things like we have to look into the diurnal escape from high temperature. For example, viable pollen and successful fertilization. Some of the papers published by Dr. Jagdish Krishna, earlier he was in Kansas State University and now he has moved to Texas. So he came out with the concept very clearly, like proof of concept, that some of the rice genotypes, they flower only in early morning, and that's the reason they are tolerant. But if you bring this particular trait in cultivated varieties, good agronomic background, it is possible to make them tolerant to high temperature. That was the factor. If that is the case, then how to phenotype them? What like imaging system, what phenotyping tools, and how you are going to design that one is very important. Then other one is carbon dioxide responsiveness. Nowadays, everyone is talking about how like to trap that carbon, how to increase the carbon stock, and how to enhance the carbon sequestration and all. That is possible. We have to design, but driving force is already achieved success. Like only one trait, if you focus, like deep rooting, then it is possible to make a drought intolerant rice cultivar into a drought tolerant rice cultivar. Very clearly proven. The work started with the concept, proof of concept, some work in the lab, and then demonstration the very much in the field condition. It was possible. That is the driving force. And here exactly we have to have a strategy. Strategy, review the mechanism, pick up the traits, and for that particular traits, you design all those machine learning algorithms or artificial intelligence and screen the genotypes. Then answer the question, which genotype is better, or what is the variation existing for that particular phenotypic trait, which will be helpful in identifying the gene and QTL. So in summary, reimagined quest for plant feature designs needs genes, and that needs the process. Phenomics and non-invasive tools can be designed and can be made affordable, but we should be careful about designing image acquisition protocol, image conversion into data protocol, and also data analysis protocol. Physiological insights needs translation for novel traits. Lot of physiological mechanism we need. You pick up one by one and design the phenotyping solution for that. Imaging scientific hints about resilience to abiotic stress, that concept will continue to evolve and support efforts for climate smart crops. That's what the optimism. With that optimism, I express my deepest sense of gratitude to all my colleagues who were part of this one, all the visiting scientists from India as well from outside countries, and all those RAs, SRFs, and YPs who are there. And I'm thankful to NPPGR and particularly Jyoti and all your colleagues for giving me an opportunity. I must thank Nikra Krida for supporting all that plant phenomics work at National Institute of Abiotic Stress Management, my previous institute, then expert work group, phenotyping of wheat, from where I get a lot of information about latest phenotyping progress. Indian Institute of Wheat and Barley Research, Karnal, where I started my career as scientist. Then International Center for Tropical Agriculture, Cali, Colombia, where I spent 3.5 years in establishing phenotyping platform for screening gene technology for rice. Then my previous institute, National Institute of Probiotic Stress Management, and my present institute, Central Institute for Arid Horticulture. You are all welcome to this institute. It's a wonderful institute to give you a glance of arid biodiversity and how it can be useful for improving the rural economy in arid region. Thank you very much. Thanks a lot.
Thank you very much, sir. So, congratulations once again for resuming charge of Director CIS to
Thank you,
become here
Jyoti. Thank
from
you,
my
Jyoti.
side
Thank
and
you.
from participant side, because they were asking that now sir has become director, so they're thinking, okay, whether lecture will be delivered or not, like that. Some persons were asking.
No, you are all welcome at any point of time. Those students particularly, please, if you have some questions, give me some WhatsApp, and then just give me a call in between 5 a.m. to 6 a.m. or 7 a.m. I will be able to answer some of the question, and we can design some of the phenotyping solutions for your work. If you are interested in coming out with your own ideas, how to do that, I can help you.
So, dear participants, if you have any doubts or any queries, you can raise. So, sir will answer now also.
Hello, madam.
Yeah, please.
Well, first, I would like to thank very nice presentation. I have some, madam, one question.
Yeah, please ask.
Madam, if you any technology for deficiency of micronutrient and pathogen infected plants, how to different? In some cases, same symptoms is there in plant, same symptoms in there in like nutrient deficiency or pathogen. So any technology regarding this? In case of citrus greening bacterium in citrus, deficiency like symptom is there, but pathogen is there. So any technology for how to identify this type of plant?
Yeah. No, that's really being addressed. Okay. So what the scientists are doing, they are collecting all the symptoms. They are making the library of all the symptoms, not in one or two, hundreds and thousands images they generate. And they just come out with some algorithms to tell that this is due to the disease or what you call insects. So when you talk about only the deficiency of nutrient, then there'll be no injury. But when you talk about these injury to the structure of the plant, in the beginning, at least in the initial stages, I'm sure that you will agree with that one. If it is pathogen or it is the insect, there must be some one or the other injury will be there. In addition to all those features that you are talking about, that can be one of the hint where we can separate the pest caused differences in plant features, particularly RGB, and only the micronutrient caused deficiency. Then other one is, always remember that unless you know the type of stress, it is really difficult to interpret about the symptoms. So whenever you want to go with the phenotyping, you should be sure about what type of stress was there even under that condition. So if you're talking about already deficient soil, or the crop production system where you have not applied any additional fertilizer at all, or the arid region where the nutrient holding or water holding capacity is very poor, then likelihood of the symptoms, for example, greenness to yellowing, will be more due to the deficiency. If it is the insect, I can capture that symptom also by engaging highly sensitive sensors, highly sensitive imaging systems. It is possible to do that one.
Thank you very much, sir. Thank you very much, sir.
Yeah.
Thank you very much, sir.
You're welcome. You're welcome.
Yeah, any another person, if they can ask now. We have time, so they can have some quick question. So, sir, AI based that modeling is also being developed, no, for this disease infestation diagnosis and all.
Yes,
in
some
in
of the
various
labs, they are
crops.
like, yes, some of the labs are like generating that set of data, and even some apps based on their library. It is possible to do that one. Yeah.
So that will also help in distinguishing whether it is nutrient based or
Yeah, yes, that's
deficit.
right. And then other one is, you remember, that's one of the very important question. When, if you say that you challenge me to tell that whether it is due to the nitrogen, the symptom, then definitely I am going to affordable phenomics like only visible camera and RGB sensors, RGB pixels and all those things. If you are lucky to get hyperspectral imaging system, then you can come out with the model which can tell you about nitrogen induced symptoms. You can predict. So you give different level of nitrogen, what exactly happening in that particular crop over a period of visible camera and also with the hyperspectral imaging system. And some of the wavelength within that hyperspectral range, all that spectral range, you will be able to combine these two things and you can come out with your own indice to tell that it is due to the nitrogen, but not due to the disease or insects. That is possible to do that. And people are going to do and come out with those tools. It is possible in years to come.
Okay. So if there is no question further, in chat box also there is no question, but there are very much appreciation that very informative presentation is there. So many people are they are sharing their views that it was very much useful and all. So then I would like to thank sir for accepting our invitation. So on behalf of myself, organizing team, and on behalf of NVPGR, Director NVPGR, I would like to thank you, sir, for accepting our invitation to deliver this very much important topic, the burning topic in the today's era, sir, affordable phenotyping, because high throughput phenotyping is still costly. So you have gave overview about the affordable phenotyping too also, so that which will be economic for the phenotyping of large number of germplasm and mapping population. So thank you very much, sir, on my behalf and then on behalf of trainees also, that you accepted our invitation and delivered such a wonderful talk, sir. So thank you once again, sir.
Thank you. Thank
So
you, Jyoti.
I
And
request
regard-
trainees. So if they have any question, they can put in the email, or as sir told that he can reply through WhatsApp also or chat also. He will help in designing the means program and experiment layout. How will we go for any problem?
Okay, thank you, Jyoti. Thanks to all the trainees who could participate in that one. There are something like 47. I'm very happy about it.
Yes, sir.
And at any point of time, if they feel, my email is there, and you can share my phone number also with them.
Yes, sir. I will
and
share,
the
sir.
WhatsApp one, so they can just ask question, and whenever they feel that they want some phenotyping solution, I can share my views and ideas. But I am very much confident that all these young guys, they have better ideas than mine, and they will be able to come out with the better tools than what I talked about. Because you see, the some of them, they were done by the MSc students, and all the trainees, they are capable of doing it. And thank you very much again. And I wish a nice time ahead for all the trainees, and wish happy moments for this Mahashivratri also for all of you. Thank you very much. I will close it here. Okay.
Yeah, thank you very much, sir.
Thank you. Thank you.
Thank you, sir. Thank you very much.
You're welcome. Thank you.
Friends, now we have next lecture in the afternoon session at 2:30 p.m. So all are requested to join. So there is one important practical about embryo rescue in the afternoon session. So Dr. Subhas and Ravi will take care of that session. So thank you one and all.
ज्योति मैम, हेलो हेलो, वो लोग खेल रहे हैं, इसका नेट चल रहा है। ज्योति मैम, रवि सर, डॉक्टर रवि, क्या मेरी आवाज सुन रहे हैं?
सुन रही हूँ।
अच्छा ठीक है मैम, आपने भी ज्वाइन कर लिया, ठीक है। अभी ठीक है मैम। हेलो हेलो, ज्योति मैम,
हाँ 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 हाँ, सुन रही हूँ।
मैंने आपको को होस्ट बना दिया है और रवि सर को भी बना दिया है।
ठीक है, सुभाष को ही बना दीजिए, सुभाष भी रहेगा
ज्वाइन कर रखा
आफ्टरनून
उन्होंने?
में। हाँ हाँ, सुभाष अच्छा ज्वाइन किया कि नहीं ये नहीं पता। अच्छा, जब ज्वाइन
हाँ
रहेगा तभी बना
हाँ,
सकते
ज्वाइन
हैं।
किया हुआ है, ज्वाइन किया हुआ है।
अच्छा हाँ, तो उसको भी बना दीजिए, ठीक है।
हाँ, अब 3 हो गए ना। कहने का मतलब है कि मैं ट्रैवलिंग कर रहा हूँ और मैं
अच्छा अच्छा,
नेटवर्क बाहर हो गया तो फिर प्रॉब्लम हो जाएगी।
ठीक है, कोई बात नहीं, फिर देख लेंगे हम लोग जो है, ठीक है।
ठीक है, ओके।
ठीक
और एक बार मीटिंग बंद हो जाए तो मेरे को फोन कर देना, मैं जो क्लोज कर दूं मीटिंग।
ठीक है ठीक है ठीक है ठीक है, ओके। हेलो। हाँ हाँ हाँ, की हाल चाल, गोर लागे छै। हाँ 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 हाँ, के सब खाली हाँ माँ ऐसे अपने साल से तो नहीं हो ने किछु ने केलक हन देबे केलखिन हन जैसे से फ्लाइट से अखन जस्ट उतरबे केलखिन हन हम कोनो मीटिंग हमर चलल छै तो ओइमे जैसे से खत्म भेलै हन अच्छा 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 ओके 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 अच्छा 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 ओके हम्म हम्म हाँ सब ठीक ठाक छै से 27 हम्म 28 के पूसा जेबै फिर 1 के वापस आ जेबै 29 के महीना नहीं छियै मने 28 महीना छियै हम्म 1 के वापस एबै तो 28 के रहबे करबै आ अथी 27 के रहबे करबै तो 2 नाइट तो होबे केलै हाँ तो 27 के तो रहबे करबै 28 के आबिए जेबै भरि दिनो तो थोड़े रहबै छै से आबिए जेबै आधा दिन में ओ कते दूर छै उषा छै से
ज्योति मैम आप अनम्यूट हैं, म्यूट कर लें।
ओके ओके
Hello, Dr. Vartika. Good afternoon.
Good afternoon, Dr. Subhash. Am I audible?
Yes, yes, you are very much audible, madam.
Okay, thanks.
You can check your video which you play,
Acha, okay.
because sometimes it is creating problem.
Okay, I will check.
हेलो
Good afternoon, Dr. Gotmi.
गुड आफ्टरनून सर, कैसे हो?
ठीक है मैडम, ठीक है, बढ़िया। और सुनाओ।
और अभी सुनाएंगे।
सुनाओगे?
कैम्ब्रिया रेस्क्यू का सुनाएंगे।
रेस्क्यू का सुनाओगे आप? चलो अच्छा है।
नहीं, आवाज ठीक आ रहा है ना?
नहीं, अच्छा आ रहा है, अच्छा आ रहा है। एक बार स्लाइड वगैरह चेक कर लेना अपना वीडियो, और जो है ना।
वर्तिका विल डू दैट।
आई आस्क्ड, मैंने फोन बोला है डॉक्टर वर्तिका को।
ठीक है।
चेक कर लेंगे एक बार वीडियो स्पेसिफिकली ना।
हाँ,
बाकी
बिकॉज वो
स्लाइड्स
स्लाइड्स में डाले हुए हैं, उसको
हम्म।
देखेंगे वो कैसा आएगा ना।
हाँ।
वर्तिका, थोड़ा सा आपका ना वीडियो सेट कर लेना। आपका TCCU team.
Yeah, TCC team.
वर्तिका का आवाज नहीं आ रहा है।
Dr. Vartika, you are not audible.
Now it is okay?
Now it's okay.
Okay, thank you.
Thank you, madam.
Happy
When you will
Shivratri.
Happy Shivratri to all.
वर्तिका, एक बार स्लाइड्स शेयर करके ना वीडियो देख लेंगे।
Slide is visible. Put in presentation
Slideshow.
mode.
हाँ,
फिर उसको घुमा भी देना।
एक बार वो चेक कर लीजिए, मूव हो रहा है, स्लाइड नहीं हो रहा है।
is it moving?
Yes, it's moving.
Fine. हमें वो चेक करना है ना वीडियो।
हाँ, वीडियो चेक कर लीजिए।
उसको
वीडियो आ गया था?
वीडियो नहीं आया।
मीन्स आया था।
हाँ, मैंने हटा दिया है, दोबारा से एक और है।
ठीक है।
वॉइस बहुत ज्यादा तो नहीं है?
नहीं, ठीक है वॉइस।
इट डिपेंड्स ऑन हम कितना रखते हैं ना।
ओके,
ठीक है, आपका तो ठीक है, क्लियर है।
क्लियर है, कोई
आप
इशू नहीं।
बंद किया?
ओके।
स्ट्रक हो रहा है वर्तिका। बंद किया था क्या आपने उसको?
तो मैं अनशेयर कर दूं अभी?
हाँ, अभी अनशेयर कर।
Dr. Subhash,
Yes,
I
madam.
think 7-8 minutes are remaining.
Yes. We will wait. We'll wait up to 2:30. Then we'll
Okay.
start.
Okay, fine. Thank you.
ठीक है सर।
Good afternoon, friends. Dear participants, good afternoon. And madam, I think we should start. In the meantime, our participant will join.
Yes, that is okay.
I think because already it's 2:30, so we should start. So dear participant, we have a very important practical lecture on embryo rescue techniques in wide hybridization, which is very important in while when you are going for a wide hybridization point of view. Let me first introduce our today's speaker, Dr. Vartika and Dr. Gautami, and I formally welcome you, both of you, madam, Dr. Gautami and Dr. Vartika.
Thank you so much, Doctor.
Let me share the slides first. Okay. Ravi, are you there?
Okay, I will share. Please wait. Please
And
wait.
my slide. If thank you, Dr. Ravi. Friends, first I introduced our today's speaker, Dr. Vartika Srivastav. She is a scientist in senior scale in the division of tissue culture and cryopreservation unit. She is working in ICR and NPG New Delhi, and her nature of work is in vitro gene bank crop curator for tropical crops like Musa species, that is specifically banana, and taro. And she is doing in vitro and cryo also in Musa for 49 accessions and in taro 96 accessions. She is also involved in the embryo rescue in linseed, specifically interspecific crosses, and pollen and dormant birds cryopreservation studies in different crops, and development of seed cryopreservation protocol in Prunus nepliensis, specifically she is developed, and she is also developed protocol in Garcinia species and taro germplasm. And she has received many awards, and among of which she has received Young Women Scientist Award in 2020, and she also has overseas experience in lab in Belgium, and she has a visiting scholar there, and she has published more than 45 articles in such as 17 research papers, 25 book chapters, 10 popular articles, 2 training manuals. Next, please. Dr. Ravi. We have another speaker, Dr. Gautami R. And she is a scientist senior scale at TCCU NBPGR New Delhi. Next, please. Next. Yeah. She is working in vitro gene bank crop, and she is a crop curator of medicinal plants, and currently she is working on in vitro conservation and cryopreservation of medicinal and aromatic plants with special reference to threatened species, bulbous crops, pollen cryopreservation, and embryo rescue. She has developed shoot tip cryopreservation for many crop species, including Bacopa manori, Gentiana kuru, then Dioscorea deltoidea, and Schwarzia cheraita. She has received many awards. She is also award and recognition, and she is a gold medalist in graduation, post graduation, and her doctorate. And she has received best publication award for her research paper, and she is also recipient of award, and also received best young researcher award. She has published more than 85 articles, including research paper, book chapters, technical and popular articles. Madam, both of you are welcome to our training program, and I request you to share your presentation or share your videos with our participant.
Thank you, Dr. Subhash. Without wasting much time, I think we should start. My slide is visible now?
Yes, madam. Do it in presentation mode, madam.
Yeah. Okay.
Ma'am, it is not in presentation mode. Yes, ma'am.
Now it is okay?
Okay, ma'am. Okay.
Okay, thank you. Okay, thank you so much, organizers, for this opportunity. I will be, along with Dr. Gautami, presenting some practical aspects of embryo rescue technique in wide hybridization. So, the major objectives of any plant breeding program is to improve the characteristics of plants so that they become more desirable agronomically as well as economically. So these are some of the parameters on which plant breeders often use to work to develop new varieties that not only produce higher yield, but that are also resistant to various pests and diseases, and also abiotic stresses like drought, salt stress, and so on. Apart from that, breeding is also going on for synchronous maturity, non-shattering characteristics, and on the maturity duration of the crop species. To obtain such type of excuse me, to obtain such type of variation among the population, we can have a genetic makeup of different type of species in a gene pool, and naturally existing variability like the plants or the species which are available in the natural environment can be domesticated. They can be collected and introduced as new varieties so that the properties or the traits of importance can be identified. And also new variation may be created by the use of hybridization techniques, mutation breeding, polyploidy, somaclonal variation, and genetic engineering. So such type of variations are of utmost importance for a breeder to find out those traits which are really of importance in this era of changing climate. So through various selection methods, as well as evaluation, multi-location evaluation trials, and multiplication of the identified superior types can be further used for release as variety or distribution to the farmers. But sometimes, since we know that some of the wild types or some of the species are not easily crossable, so the concept of gene pool needs to be understood at that point of time, because we can say some species which are closely related can freely intercross, and they can produce viable plants, and those plants produce fertile seeds. But some of these species, and these species which can freely intermate, they fall into the category of GP1, that is the primary gene pool. So such group are fertile are known as primary gene pool 1 species or varieties. But when some of the species which are closely related to the species of gene pool 1, but they produce some of the fertile crosses in F1s, not most of it, but some of the fertile crosses can be achieved. So such species or varieties or closely related species are grouped under the secondary gene pool category. Then comes the tertiary gene pool. These are the distantly related species, more often including the wild crop wild relatives and other species that can intercross with GP1 and GP2 species, but the hybrid produced after the crossing are anomalous, lethal, or completely sterile. That means they will either not produce flower or they may not produce a viable seed. And thus the gene transfer is not possible, and it requires additional measures such as embryo rescue or chromosomal doubling to obtain a viable plant. So basically, distant hybridization is of can be of two types. That is, when the crossing is made between two different genera, it is called as intergeneric. And when we are crossing to different within the species, then it is known as, between two species, it is known as interspecific cross. You might have heard this term pre-breeding, and you might have attended the lectures previously on pre-breeding. I'm not going into the detail, but one very important thing about pre-breeding is that we are using here the crop wild relatives. So the crop wild relatives may be harboring they are the source of some of the important genes which we might have lost during the domestication and selection of the cultivated types, of high-yielding types. So these crop wild relatives, sometimes they are very far distantly related, that they are not being able to produce a viable seed after the breeding program. So basically, the pre-breeding program aims to introduce such crop wild relative into breeding lines and to produce pre-bred lines which can easily hybridize with the working collection. And this is now very much, you can see here, for example, in wheat, if somebody wants to do a pre-breeding experiment, they may have a huge number of traits on which they can work. But when we talk about distant hybridization and pre-breeding, there are certain barriers. These barriers pose problem. They are the external as well as the internal barriers, because of which a fertile hybrid is not produced. And when we understand these barriers, we can implement the strategies to overcome it. For example, the external barrier can be spatial and temporal isolation. That means the male and female plants are not located at the same place, or they may not flower at the same time. So these are the barriers in which you cannot conduct crossing program at the same time or at the same place. So such type of barriers can be handled by different strategies. However, the pre-fertilization barriers, for example, the non-addition of pollen germination on the stigmatic surface, or lack of stigma receptibility, and lack of pollen pistil compatibility, or the failure of fertilization are some of the pre-fertilization barriers which may not be these pre-fertilization barriers are posing problem for the male gamete to reach to the female gamete to fertilize it. That is how the fertilization may not take place. So there is no point of formation of a zygotic embryo. However, the internal barriers, that are the post-fertilization barriers, here the fertilization take place, but sometimes the hybrid is not produced, the embryos are not produced. Even if they are produced, they may die because of the maternal effect, or even if the embryos may be alive, they may not develop into a complete plant. So these are certain things that we should know and we should understand before undergoing any wide distant hybridization program. And in this, we have various tissue culture techniques that we can use to overcome all these barriers. And in this slide, you can see that if a normal hybridization process is going on, then how the sequential, you can say the channel is after pollination, the pollen tube hydration and germination in the stigma occurs. And then after that, it penetrates through the stigma. And then the pollen tube grows into the style and reaches to the ovule, and it pollinates the female gamete. And then the embryogenesis happens, and formation of embryo, endosperm development occurs. And later on, after the completion of the seed formation, the plantlet development occurs, and after that, reproductive phase comes. But when we talk about hybridization, which is not very closely related, or you can say when we go for pre-breeding or when we go for wide hybridization, we can see at various stages, there are pre-fertilization and post-fertilization barriers. And for example, if the pollination occurs, but the pollen is not hydrated, it may not form a pollen tube. It may not stick to the stigma. And then if it not sticks to the stigma, there is germination failure. So again, this is a pre-fertilization barrier. However, even if the pollen sticks to the stigma, it extends its pollen tube. It may not penetrate inside the styler region because of the lack of stigma receptivity. Again, this is a pre-fertilization barrier. And later on, if it goes through this style, there is a pollen pistil incompatibility, or if it is a failure of fertilization, it may not reach to the ovule or to the female gamete. So these are the pre-fertilization barriers. However, when the pollen tube enters into the stigmatic surface and then it reaches to the embryo sac and fertilizes the egg, there may not be complete proper embryogenesis taking place. So there is a chance that the embryo formation may hamper. A very rudimentary embryo may form, or even if the embryo is formed, it may get aborted because of the maternal tissues, and so on. If there are possibilities that even if the embryos are produced, they may not develop the vegetative the plant may not develop properly, and it may die soon during its vegetative growth. And even if it is surviving the vegetative phase, it may not produce the viable flowers or viable seed. So these are some of the post-fertilization barriers. So the pre-fertilization barriers can be overcome by in vitro pollination and fertilization techniques. However, the post-fertilization barriers can be overcome by the embryo rescue, and it can be ovule culture, ovary culture, or embryo culture, depending upon the organ used. So when we talk about embryo rescue, they're specifically used for the inherently weak embryo that might not survive to become viable plant. And the basic requirement is to place those embryos into artificial nutrient medium aseptically that replaces the endosperm, and allowing the embryo to continue its growth and development. And depending upon the type of organ you are culturing, it becomes embryo culture, ovule culture, or ovary culture. In this slide, it is very much visible that in slide number A, you can see that the embryos are very rudimentary. The embryo is very small. It is in globular stage. However, in the slide number B, you can see this, this ovule is blackened, and the embryo inside this ovule is black, and it is dead because of the hybrid-lethality. Because of the maternal tissue, it could not grow further. However, in the third slide, slide number C, you can see a well-developed embryo inside an ovule. And this is how you can understand that what is the difference between embryo rescue and embryo culture. So embryo culture is a technique, and it applies to all type of embryos, whether they are rudimentary, they are well-developed, and it is a simple thing to understand this thing is that the embryo culture is a technique in which the embryos are cultured into the artificial nutrient medium aseptically. However, the embryo rescue term is particularly restricted to the cases where the embryos are rescued, and even if when they are not rescued, they are endangered and would not form seedlings. But mostly, these two terms are used interchangeably. So what are the different applications of embryo rescue? We have already seen through in our previous slides that embryo rescue is of utmost importance for the wide hybridization. But apart from that, we can also recover maternal haploids, and also we can hasten the seed germination and overcome seed dormancy in certain cases. And it is also helpful for the shortening of breeding cycle, as well as to achieve the seedlessness in some of the horticultural crops. Coming to the practical aspects, when we are undergoing any wide hybridization and we suspect that the embryos which we need to rescue had to be previously, they need to be rescued early, as well as they need to be they should be given proper treatments, and how to increase the chance of the survival of the hybrids. So there are certain factors that one must understand prior to undergoing the wide hybridization program. So apart from selecting the right type of parents, there are certain things which one should also consider. That one is most important is this correct stage of embryo rescue. So it is one of the most important factors where you know that at certain point the embryo is either abort, or it may not develop beyond that stage. So if the embryos are very small and they are not growing beyond a certain stage, you need to identify that stage and isolate that embryo on a priority basis. Otherwise, the embryo may die inside the embryo sac, and you may get a completely non-viable embryo during your culturing. So the identification of the correct stage is very, very important. And how you can identify the correct stage? By manually looking at the embryo under a microscope at different days of pollination. Like if you have pollinated or made the cross at the first day, you have to count the number of days after pollination and then collect the fruits and then see it under the microscope, that how on which stage the embryo is visible, whether it is a globular stage or a torpedo stage, a heart stage or torpedo stage, or a mature stage. And if you find that, okay, this is the stage at which the embryo started degenerating, you should isolate the embryo from the embryo sac prior to commencement of that phase. So generally days after pollination are counted to see the developmental changes in the hybrid embryo. Another important factor is the composition of the culture media. We know that embryos are the rudimentary structures, and they undergo a sequential developmental stages, and they are capable of forming a complete plant. But if the embryo is in a younger stage, that means if it is in a globular stage, it may not be having the root and shoot primordia defined. So they are known as heterotrophic stages, where they are totally dependent upon the maternal tissue or the endosperm tissue for their nutrition. And if at this stage you need to isolate the embryo, you have to provide them a complex media containing supplements in the form of vitamins, amino acids, growth hormones, and some of the natural extracts like tomato juice or coconut milk or coconut water. But it depends upon the species in which you are going undergoing this embryo culture or embryo rescue. Then there is an autotrophic stage in which, and that is the stage when the embryo is having a defined root primordia and the shoot primordia, and it can survive on a simple nutrition, a simple media without much addition or supplements of vitamins and minerals. And those stages are like the torpedo stage, or maybe the fully formed embryo can be transplanted into a simple MS media with sucrose, so that they may form a complete plant. And there are so many medias like White's medium, MS medium, B5 medium, NN medium, niche medium that can be used for different crops depending upon their nutritional needs, and that to be looked upon before undergoing any such type of exercise. Then the most important step, one of the most important step is to clean the surface of the explant. Most obviously, the explants are the fruits. They may be pods or siliqua or the capsules, and you have to bring those capsules to the laboratory. And the soil particles, since you have conducted these experiments in field or maybe the glass houses, so this is very obvious thing that the soil particles are present on the surface, and these needs to be removed by the use of surfactants like T-Pol 220 or maybe cetrimide. And after the removal of the soil particle, you have to continuously stir the explant into the surfactant solution, and then rinse it with the double distilled water, and then bring the explant to the laminar airflow, and then you can treat it with the various surface sterilizing agents like sodium hypochlorite, ethanol, mercury chloride, etc. And never to forget to give them a quick wash after the treatment with double distilled autoclaved water 3 to 4 times to remove all the extra particles of the sterilant. And also the duration of the exposure of the sterilant is very important, and it must be looked that the age of the explant is very important, and the concentration of the sterilant needs to be standardized based on the its mode of action as well as the age of the explant. And care should be taken so that the explants are not damaged, because they are very delicate, and if you will damage the explant, you may not be able to extract a viable embryo. Again, the culture conditions need to be aseptically maintained, and the temperature requirement of the crop needs to be understood before placing the petri plates into the growth chamber. So if you are working with the low temperature loving species, you have to put it in the lower temperature BOD, or maybe you can use the phytotrons to mimic the temperature requirement of that species, particular species. And care should also be taken while extracting the embryo that no physical damage should be made to the rudimentary embryo. Otherwise, you will end up with the callousing. It may not form the plant completely. So these are some of the points which you need to be very careful of. And now I would like to request Dr. Gautami to discuss the methodology of this technique. Dr. Gautami, please.
Yeah, thanks, Vartika. Yeah, next slide, please. Yeah, as you can see from this slide, this is the generic protocol for embryo rescue, where after hybridization, crossed pods or fruits or capsules should be collected at appropriate days after pollination, depending on the species, followed by surface sterilization in the laboratory. Later on, embryos should be excised carefully and inoculated on appropriate media. Once the embryo develops, in vitro multiplication media should be standardized for further experiments. One of the important steps in embryo rescue is confirmation of hybrids, and also sometimes we have to go for colchicine treatment in cases of F1 sterility is already reported. Yeah, next slide. Next slide. Yeah, these are the representative images of few materials required for the embryo rescue techniques. Next slide. Next slide. Yeah, Dr. Vartika has already highlighted the different crossability barriers. In the coming slides, we will discuss about few solutions for those barriers, taking few case studies that are standardized in our laboratory. One of the most common challenge faced by the breeders during hybridization is spatial and temporal isolation. And wherever possible, usually we go for staggered sowing to overcome asynchronous flowering. However, in many cases, it is difficult to attain asynchronous, means it is difficult to attain synchronous flowering. In such cases, we can go for pollen cryopreservation for long-term conservation without losing its pollen viability. By using pollen cryopreservation protocol, we can overcome few challenges of hybridization, like we can supply pollen constantly and we can overcome asynchronous flowering. Also, in the current scenario of land degradation, often we face challenges of scarcity of research plots to grow a large number of male and female parents together. In such cases, in one season, we can go for growing desirable male parents, and we can collect and conserve pollen for further use in hybridization. Next slide. This flow chart represents the stepwise procedure of pollen cryopreservation. As you can see from this slide, first we have to collect freshly opened flowers from the healthy plants from the field. In the laboratory, we can collect pollen directly by tapping, followed by desiccation in the silica gel, or in cases of sticky pollen, we can use cyclohexane method, where anthers are placed in 5 ml of cyclohexane for 50 seconds to 1 minute. Then the suspension is drawn using syringe, as you can see from this slide. Later, press the syringe and pass through the suspension through the millipore filters and collect the disc containing pollen. After this, the collected pollen is placed in aluminum pouches, followed by placing in the cryovials. Later on, cryovials with pollen are placed in the liquid nitrogen till further use for a hybridization purpose. Yeah, next slide. As you can see from this slide, we cryopreserved 24 accessions of Abelmiscus moschatus in one season and used for hybridization with Abelmiscus esculentus. Yeah, next slide. In coming slides, I'll discuss embryo rescue of interspecific hybrids. Now you all well aware of activities of each division of NBPGR. Our colleagues from evaluation has faced a few challenges in obtaining successful hybrids of mung bean and rice bean. To obtain F1 hybrids, we have attempted embryo rescue, and we could able to successfully rescue them in vitro, as you can see from this slide. Next slide. As I said in the previous slide, the crossed pods of mung bean and rice bean used to drop at 3 days after pollination. Initially, we have tried applying growth hormones containing GA3, NAA, and chitin at the base twice a day. However, we could not able to get the matured parts. Rather, we could able to prevent early abortion of the embryos. Next slide. After that, 7 days after pollination, by applying growth hormones, we collected parts and brought to the laboratory and followed a series of surface sterilization steps to minimize the risk of contamination. We washed the parts with T-PAL for 5 minutes on a magnetic stirrer, followed by washing with distilled water for 5 to 7 minutes. And pods were further treated with 0.1% bevistine and 0.1% streptomycin for 3 minutes, followed by washing with distilled water for 3 minutes. And later, pods were sterilized with 0.1% mercury chloride for 3 minutes, followed by rinsing 5 times with sterile water to remove the adhered extra chemicals on the pods. Next slide. Yeah. After sterilization, embryos were excised from the parts carefully, as shown in these figures. Care to be taken and extra care to be taken that embryos are not damaged during isolation. Then the isolated embryos are further inoculated and media. We used MS media supplemented with casein hydrolysate, coconut water, and low concentration of BAP. Then after inoculation, the embryos are incubated under standard culture room conditions as shown in this slide. Next slide. Once after the development of embryos, we may need more explants or more material for further experiments like conformity of hybrids, rooting, and ex vitro establishment. Thus, we should standardize a media for in vitro multiplication or elongation to obtain sufficient explants for further studies. In our study, we tested different media for in vitro multiplication, that is MS medium supplemented with different concentrations of BAP, NAA, kinetin, and a combination of BAP and NAA. And we obtained multiple shoots on MS medium supplemented with BAP and NAA. Next slide. Sometimes we get rooting on the same multiplication media, but sometimes we have to standardize a different media for in vitro rooting. In vitro rooting of F1 hybrids of mung bean and rice bean was a challenge for us. Though we obtained a roots and different media, as you can see from the left side of the photo, these roots were not suitable for ex vitro establishment. While as you can see from the right side photo, this yielded a good success in the ex vitro establishment. Next slide. Yeah, then later on, once we obtain a good rooting, in vitro rooting, we have shifted our F1 hybrids into the field by following the protocol as given in the slide. And also here I would like to mention one of our experience that sometimes we get F1 seeds and after further growing, there might be a chances of F1 sterility. So we cannot further pass to the next generation. Similarly, our colleagues also faced a similar challenge where F1 hybrid seeds were germinated and developed into a healthy plant with intermediate nature of parents. But the F1 plants fail to produce flowers. But for a breeder, a single F1 seed is more precious like a diamond. So in order to protect this plant, we attempted a in vitro propagation of this F1 plant using a nodal segments as explant to protect this plant without being lost. Then later on, we are planning for colchicine treatment for chromosome doubling so that we can get viable F1 plants in the field. Yes, thank you. Vatika, you can take on from now onwards.
Thank you, Gautami.
Yeah.
Now I will be discussing the work done by my student for reducing the breeding cycle of the papaya by embryo culture. So here you can see that the immature embryos were taken, the immature seeds were taken to isolate the embryos at different maturity. And we could able to reduce the crop breeding cycle by around 5 to 5.5, 5 months to get viable plants, and they are fruiting now. There is a short video prepared by Mr. Bhupendra on this aspect. So this is our culture room, in vitro repository of tropical crops. So after a few days, the embryos are exposed to natural light. The preparation for the hardening. So because of embryo culture technique, we could reduce the duration of the crop. It was reduced to up to 5 to 6 months, and which is a very, very considerable duration for any crop breeder to perform different type of or execute different type of experiments. So it is one of a kind of a study which was done by, it was just shown to explain that the embryo culture technique is not only important in case of distant hybridization, but it can be used for the different purposes to meet out different purposes in the breeding experiments. The third case study is related to the interspecific crosses made in linseed and the use of embryo rescue technique for certain crosses which are not fertile in case of linseed. So with the objective to introduce certain genes from the crop wild relative of Linum grandiflorum for bird fly and alternaria blight tolerance, we attempted the cross between the cultivated variety of Linum usitacissimum with the Linum grandiflorum. But in nature, whatever crosses we have made, it resulted into shriveled seeds. And when we looked upon these seeds closely under a microscope, we could find that the seeds were harboring a large size of embryo, but the embryo was completely dead because of the lethal gene actions from the maternal plants. So we tried embryo rescue technique to develop some viable plants in this cross. So as you can see here in this diagram, the female plants of Linum usitacissimum were crossed with the Linum grandiflorum, and the hybridized capsules were harvested at different stages of maturity, from 7 to 9 days after pollination till 16 to 20 days after pollination. And we could see that the immature capsules were having globular embryos, and the half mature capsules were having green and torpedo shaped embryos, whereas the mature capsules, when observed under the microscopes, they were having black embryos, which were shriveled as well. So we tried both ovule culture as well as embryo rescue to see whether these ovules may help the underdeveloped embryos to grow into a complete plant. But here we can see, when we observed that when we used ovule culture, all the embryos present inside the ovules after a certain period of culturing turned black, and they could not grow into a complete plant. So we hypothesized that the female or the maternal tissues or the maternal tissue is causing producing certain metabolites which are causing the death of the embryos or the abortion of the embryos. When we observed the capsules closely under a microscope in after 10 to 15 days of pollination, we could isolate the torpedo-shaped green embryos that we cultured into modified MS media, and we could get some of the plantlets regenerated. The percentage was very less of the embryos present in the hybrid capsules and the number of plants which survived. So there was no point in culturing mature capsules, but we tried some of the dead embryos also, but they did not lead to any plant production, which was very obvious. So that was also we are expecting and we are getting in the cross-pollinated capsules which are left in the field for maturity. So whatever plants we obtained, they produced quite good shoot system as well as root system, and they were hardened in a phytotron facility. Here in this slide, you will be able to understand at what stage we have harvested the embryos from the ovules. Since the embryos from 7 to 9 days after pollinated capsules were very small, they produced only callus. We tried to harvest the embryos, isolate the embryos from the 10 to 15 days after pollinated capsules, and we could get some of the best results, and we have obtained a good number of plants that are multiplied and then hardened in the maize chamber. This is a short video to describe the procedure. All anthers must be removed from the female flower, and the flower should be bagged immediately. Male flowers are harvested and the petals are removed to expose the anthers. And carefully the female flower is opened without damaging the stigma and style. The pollen are visible on the stigmatic surface. You can double check it visually. And then proper labeling should be made. And again, the flower should be bagged. So after the inoculation, the Petri plate needs to be completely sealed with the parafilm, and they should be covered with aluminum foil and properly labeled. All the Petri plates individually should be labeled to avoid any mixing. In such crosses, we need to be very, very careful with each and every pod, and they should be placed at standard culture room condition required for that particular species. And the aluminum foil is removed carefully, and then you will be able to see am I audible?
Yes, madam, you are audible.
Okay.
Very much audible.
So then these are transferred to a much simpler media, where the the basal MS media, and in test tubes also, to help them to grow its shoot as well as roots. And then after the complete root formation, they may be hardened in the net house or mist chamber, whatever facilities you have. We have hardened our linseed in phytotron because it is a low temperature loving species, and it is a temperate crop. And if we would have hardened it in mist chamber, they were all like died. So you need to be very, very careful in designing all these steps and configuring all the temperatures throughout the crop cycle to mimic its original growth and nutrition requirement. And then when the plantlets are hardened, you have to be very, very careful to see if it is producing flowers. And if it is producing flower, is it producing a complete viable capsule or not? So we have encountered the drying of the capsules in this F1 hybrid. And just after knowing that this is happening, we have again started our program to remove or isolate these capsules from the plant when they are still green. So we are under the process of regenerating the embryos from these hybrids as well. So this is how this embryo culture technique is helping us not only to recover the distant crosses, but also otherwise infertile process we are also able to save, or you can say we are able to reduce also the crop cycle as well. So rather than waiting for the complete capsule to mature, or rather than thinking that, okay, the capsule was now dried, so what we will do, we will change our strategy. We suddenly went and we again executed all the sort of experiments which are required, which we have done previously to save those explants. So we are now having some of the hybrids, F1 hybrids, live with us, although some of the these capsules are dried. So we are hoping for the best, and this is how this is the beauty of this technology, that we are now having the F1 which are naturally otherwise infertile, or you can say may not produce a complete plant or may not produce a viable capsule. So in conclusion, I would rather say that these innovative tools and technologies are very, very important to facilitate breeders to develop new varieties with desirable genes, to face the issues of climatic change. And this technology, embryo rescue technique, greatly aids in breeding programs, most of the pre-breeding programs recently. And this is how we can use the distantly related species and incorporate favorable genes quickly and effectively. And for different crops, we need different protocols, and these protocol needs to be refined periodically depending upon the crop need and the breeding problems that we face on the ground. And such optimization requires continued innovation and methods, and also in technologies. With these words, I would like to thank Director ICR NBPGR, DBT for funding, our collaborators from different centers, officer in charge TCCU, and our technical and supporting staff of TCCU They are very, very helpful. They are very, you can say, experienced. And without them, we could have not achieved a single milestone. And nevertheless, I would like to thank Ms. Kshitija Shirbhate, who is project assistant, DBT project, Vijay, SRF in the project, Raghavendra, and my student Bhupendra to help us prepare the videos which we have shown today. Thank you so much. And last but not the least, I would like to thank the organizers to give us this opportunity from my behalf as well as from Dr. Gautami's behalf. We are very thankful that you have given this opportunity. Thank you so much.
Thank you very much. Such a nice presentation. Now, I would like to ask our training participant to share, to interact with our speakers. And if you have any question, please ask. Madam, one question from Dr. Mohammad Makbul from Squest. There is one question. Embryo rescue is reported in case of any tree species. You can see the question also.
Yeah, I'm looking at it. Yeah, there are many reports. Embryo rescue is often used, but I don't think he's asking about fruit crops or maybe something else. I mean, if it is a
Muhammad, you can directly interact with our speaker. Dr. Muhammad, are you there?
Yes, sir. Yes, sir.
Yes, you can ask your query.
Yeah, good afternoon. Actually, I want to know about the tree species, forestry tree species, whether such technology is used there or not.
I'm not aware of it, but we can use it. But the problem is that these are slow growing and woody tree species. So the results are very slow. But definitely you can use this technology in tree species, but you have to see for what purpose you need to do this type of hybridization and whether actually you need embryo rescue or not. Actually, embryo culture is often confused with embryo rescue. Embryo rescue is only required when the embryos are lethal and they are not going to produce a viable plant. And if the plant is, if the seeds are viable and you are able to get the seeds itself in the after F1, after crossing, the F1 is viable, you need not have to go for a embryo rescue technique. So it may depend. I'm not aware of any tree species which is being, where this technique was used. But yes, for the crop plants, we are often using it.
Okay, okay, thank you.
Another question, madam, from
Hello.
Yes,
Hello,
you can
sir.
ask.
Madam, how many years pollen survive under cryopreservation?
Yes, this is a very nice question. In fact, not only pollen, all the viable propagules, be it shoot tip, be it seed, be it embryo, if you have conserved it for, let's say, for example, there is a pollen in which the pollen germination or the in vitro germination is 80 to 90%. And after cryopreservation of 1 hour, or maybe let's say 24 hours of LN treatment, you have retrieved that pollen from the cryo tank, thawed it properly, and conducted in vitro germination as well as in vivo pollination. You will be 100% getting 80 to 90% pollination or germination survival. Pollination and survival in case of seeds or different propagules. So theoretically, you can conserve it for n number of years, like 100 years, 200 years, or maybe even more. So theoretically, it is so, and we have in our gene bank pollen conserved for.
In practically, madam, in practically.
Yeah,
Practically also. Yeah, sure.
Yeah, so far for the hybridization program, so far recently we have done for a 6 months, we have conserved Lathyrus pollen for 6 months, and we have used for hybridization and we got success. So far we have tested for the 6 months, and in the okra we have tested for 3 months. As a theoretically, it has been said that n number of years. And it has been reported with other seeds, other propagules like seeds and in vitro shoot tips. So same it is applies to the pollen also. In our tissue culture unit itself, we have tested post regrowth of in vitro shoot tips after 10 years of cryopreservation, and we are getting same regrowth. Only plus or minus without significant differences being found. Yeah, thank you.
Madam, and success rate
Yes,
under hybridization?
yes, success rate already we have a published literature that there is no effect of pollen cryopreservation either on fertilizing ability or on seed. We also tested that we obtained seeds which we used pollen for means hybridization, and we tested its seed vigor and we compared with the parents, and there is no difference on its performance. Rather, seed vigor index has increased after using cryopreserved pollen. So this is accepted recently, our Lathyrus paper. So we will
Thank
share
you very
with
much,
you.
madam,
Yeah.
for nice presentation.
Okay, thank you.
I have learned so many things, madam. Thank you very much, madam.
Thank you.
Madam,
Thank you.
Madam, there is one question. Can we use this technique for lentil crop? From Dr. Reena Mehra.
Yeah, means you can use which technique, whether it is embryo rescue or pollen cryo. Anything you can use.
Embryo, I'm asking about embryo rescue, madam.
Yeah, you can use the technique, but few modifications is required based on your crop. So that you have to do experimentation at your end.
Okay.
Then another question by Dr. Devrajan. In case of mung bean and rice bean crosses, the F1 will be normal fertile hybrid, or we have to go for embryo rescue?
So as I already said during my presentation that our evaluation scientists have faced a challenge of means abscission of crossed pods after 3 days after pollination. So we have attempted embryo rescue because of that reason. So it is already reported that there is a pre and post fertilization barriers between mung bean and rice bean, and you can get few seeds, but again passing through the generations with that seeds also important. For that we need to attempt more number of crosses. As a breeder you can know that we need, if we want to get a few seeds, we need to attempt many number of crosses. Yeah.
Any other question?
Something is there, Malarveli, but it is not visible. Is that the same question?
Dr. Piasi is said something. You can see Dr. Vartika.
Yeah, I'm just looking at it. Thank you, sir. Thank you so much. Somebody asked that whether we can use the ready-made media. Yes, you can definitely use, but it becomes a little bit expensive to purchase ready-made mixes.
Yeah, Vartika.
Yes, Dr. Vyasi said that it's like a dream come true for linseed breeder.
Yes, sir. It is very, very difficult. It was very difficult. In fact, the F1s are not, producing fertile, capsules. They are drying up. So it is very difficult. In nature, it is not at all possible. There are so many papers that states that linseed, Linum eucetacissimum cannot produce viable crosses with grandee. So yes, it is.
And there is a question from Vishnu Gite, Dr. Vishnu Gite.
Yeah. As I said, initially we have tested application of different growth hormones for embryo development and all, but unfortunately we didn't get success in this in contradictory to the previous reports. So that's why we have attempted embryo rescue. So this growth hormone application we can try for the different crafts. Sometimes it will be successful for those parents, sometimes it will not be.
Hello.
Yes, sir.
Madam, one question. In Sesame, madam, we have so many crosses, but success rate is very low. So any technique for increasing the success rate of Sesame crosses during F2 generation?
Yes, Dr. Gautami, would you like to answer?
Yeah, you can answer.
In
Any
Sesame,
technique
we have not,
because
actually, we have not practically experienced this in Sesame, but we can discuss it later on. We can help you and we can discuss what are the different challenges that you face. You can have our email ID, we can interact and we can find out what can be done.
Right,
Actually, recently under DBT project, we are trying for embryo
we
rescue
have also
with
part of
sesamum.
DVT project. Madam. We have also part of DVT project in Sesame.
Yeah. So we are
Yeah.
trying, sir. I am involved in that, and this time I have attempted embryo rescue of sesamum hybrids, but so far it's in developing stage only. That's why
Right.
I can't comment on that. But we can discuss it together.
Right, madam. Right.
Yeah,
Thank you very much, ma'am.
Thank you, sir. Thank you.
Thank you.
Any other question? Friends, if you have any question, you can ask. Even you can contact with Dr. Vartika and Dr. Gautami at any time. They will be very happy.
Yes, we will
to
be
share
very
with,
happy.
you their knowledge.
Yeah.
Definitely.
Because we are also learning in a learning phase. This is the first time we are doing it. We have not got any type of training on this.
Yes.
Whatever we are attempting and visualizing, we are observing, and then we are sharing our knowledge with each other, and we are trying to move further. So we are also in learning in stage, and we would like to collaborate with those who are actually in this field and would like to collaborate and do some good research work. They are most welcome. Our email IDs are available on our websites, and you can always contact us. Thank you so much.
If there are no question, then I may propose a vote of thanks, formal vote of thanks. Anyone, any question, any query?
Yeah, sir, Dr. Subhash. First, I would like to thank all the participants for such a interesting interaction with us. So definitely we will be helpful always, and you can contact us for any more doubts through our email. Thank you, everyone. Yeah.
Okay, thank you very much, madam. I on my behalf and on behalf of organizing committee, I thanks both of you for delivering such a the wonderful practical lecture on embryo rescue technique in wide hybridization. And madam, both of you beautifully explained different techniques for wide hybridization and their barriers there are, and overcoming these barriers. The videos you shown on the in vitro establishment of papaya plant from seed embryo and embryo rescue in linseed is very really commendable. And madam, certainly our participants are benefited from your highly informative lecture. Thank you, madam. And you interacted very nicely with our participants.
Thank you, Dr. Subhash. Thank you so much.
Thank you, madam. Madam Dolava, sorry, thank you.
Thank you, sir.
Thank you, everyone. By patient listening of lectures. And we will meet on Monday then, 9:30.
Okay, sir. Thank you very much.
Okay, thank you very much. Thank you, everyone. Thank you,
Thanks,
madam.
Gautami.
Be with us.
Thank you, Vartika.
Both of you.
Bye.
Okay. Dr. Ravi, are you there? No more money water. Hey, do ठीक से चलो मैं ना